Good morning, everyone. First of all, if audio and video both are working fine, let me know, please. Wonderful. So, yesterday we had stopped at understanding the basic understanding of what cloning is. We understood the process, we understood the definition, and this is where we are going to begin from. Now, what we understood is what the process of cloning is and how exactly animal clonings, etc., have happened in India and otherwise in general. Now, what we see is, as the next topic, is the concern that we have over human cloning. That what we understood is that animal cloning examples have been seen in India, and of course, there are examples in the world also. But when it comes to human cloning, we see that largely it is banned. Largely, it is not allowed by any government in the world, including India. Now, why exactly is that? The reason less is less scientific in nature and more societal in nature, because we see that there could be a lot of difficulties that the society might face, and there could be a broader change in the way society functions if human cloning is allowed. First, that's why if you look at the first reason, it might undermine society's respect for human life, because now what you are doing is that you are capable of making a new life with the help of cloning. We are able to replicate a life with the help of cloning. So this is something that goes beyond the natural process of reproduction and how the entire society functions and the kind of value that we give to life because of all that. But with regards to cloning being a reality, this can change, and this is something that we are afraid of. Now, at the same time, what we also might see is that they it also might lead to the clones being treated as secondary race or even as slaves, that everyone in, or let's say any person who is born out of cloning, might not be seen in the same light as the people otherwise. So that is again a concern from the society's perspective, that this might divide the society into who is a natural born and who is a clone. So that also becomes one of the challenges. And not only that, when we are talking about the society, this goes one step further, that it might also undermine institutions like marriage and starting a family, and uh, all the social parameters from that perspective. Because ultimately what happens, that the single parents also know that they are now capable, because of human cloning, they can also become parents without needing the other side. And in this is the scenario where it might undermine what marriage is and why marriage is required and all those things. So that also becomes one of the challenges, that it might completely change the fabric of the society as it exists today, and we do not know what direction it can take over a period of time. And then it might even create a global security concern, that maybe there are organizations today or even governments around the world, they might start thinking of creating an army which is made of clones, and maybe they start that function today thinking of it, and maybe 10 years later, 15 years later, they have a cloned army ready with them. So all this right now looks like a science fiction, but it is not something that which is not a reality, which is not possible in the coming days. So that's why we need to understand that all these things may not be real right now, but this is where, I mean, every science of tomorrow stems from the science fiction of today. So that's why if it is not looking to be a reality today, it does not mean that it might not be reality tomorrow, or maybe 5 years or 10 years from now. And then there is this concept of designer babies, and human cloning might also lead to this concept of designer babies. Now, designer babies, basically, we are, in case of designer babies, we are talking about children who are being born, where we are taking care of what genes we want in them, what genes we want in them, what characters we want in them, and we are ensuring that all those characters and genes, they are present in the child. So that's why the term designer baby is used here, because we are designing that child according to what I need. So same goes for when human cloning is tried, that it's not that cloning will happen from a person, a regular person. Maybe the demand for cloning happening from people who are, let's say, famous, or people who are well established, whether in academics, whether in industries, whether in entertainment. So wherever they are, and maybe people want that their children should look like them or their children should be like them, and then in that case human cloning might take a turn in that scenario. So that also is a concept which becomes a reality with regards to human cloning. So that's why there are all these concerns that we have to think of when it comes to human cloning. Similarly, one more thing that we need to understand, and a couple of you had asked also yesterday, that is cloning same as surrogacy? The answer is no, because when it comes to surrogacy, the entire process is very different. The outcome is also very different, and that's why we cannot compare these two when it comes to the entire process. So for example, in case of cloning, what we discussed is that the entire process of cloning starts from a somatic cell, where you take the somatic cell and you take out the nucleus of it, and then you insert it inside an enucleated egg cell. So that's very different from surrogacy, because surrogacy in process, or what you call as in vitro fertilization, or the concept of test tube baby, these concepts are the same. Surrogacy or in vitro fertilization or test tube baby mean the same thing. And in all these cases, this is where uh, the process is still sexual in nature, because there will be fertilization that will happen between the male and the female reproductive cells. So that's why as a process there is a mixing of genes that happens in case of surrogacy, whereas in case of cloning we have understood that it's only one parent who contributes to all the genetic material for reproduction. So that's why process wise it is different. Similarly, so that's why when it comes to cloning, we know that somatic cell is taken and the child is born out of a single parent. And in case of in vitro fertilization, an egg is fertilized by the sperm transferred to the uterus. So that's why the process itself is different. One can be called as sexual reproduction and the other one is an asexual reproduction. Then, so in case of surrogacy, what do we do? In case of surrogacy, there will be male and the female reproductive cells which will be involved, and then the fertilization will happen, and embryonic development will happen inside the uterus of a surrogate mother. Then uniqueness of progeny, because in case of cloning, we discussed that same genes, only one set of genes are being replicated. So that's why the progeny will be an exact replica of the parent, exact replica of the single parent from where the child is born. And in case of surrogacy, what we see is that here, um, because there is mixing of genes, there will be uniqueness in the progeny, and that's why the progeny will have the characters from both the mother and the father, and not only replication, which was the case with cloning. Then one major concern has been about lifespan, something that we also discussed yesterday, that if you look at the lifespan of the animal clones, you'll find that animal clones haven't lived for very long, right? They haven't lived for very long, they have not been able to sustain. And even if they have been, we have had to keep them under very strict conditions, very ideal conditions, so that they can survive, because they do not get the natural characteristics of survival that usually in natural process of reproduction one would get. So that's why their lifespan is also a challenge, that they do not lead a normal healthy life, and until now we haven't been able to do that. So only in few cases we have seen that of late we have been better with animal cloning compared to whatever we have seen until now. And then there are ethical concerns. In case of cloning, there are ethical concerns, because we discussed about all these things, the problems with the society, the problems that it can create. Apart from that, one more aspect you also have to understand is that the entire work in cloning happens on the embryos, right? The entire work happens on the embryos, and the same embryo would have developed into an individual, would have developed into an animal. So while you are doing the research on embryos, and if let's say it is not being used for reproductive cloning, it is only to be used for therapeutic cloning, then in that case there is another chance that we are killing the embryos. A lot of studies are doing nothing, a lot of research are doing nothing but killing those embryos, which is also questioned because of the fact that this is equivalent of killing a life which could have been born later. So that's why there are ethical concerns also which are raised with regards to human cloning. So there are all these issues that we have with cloning altogether. One question I would want you to see. This is a question that was asked in your paper. I'm launching the poll for you. Wonderful. 90% of you have marked the correct answer. So we are talking about somatic cell nuclear transfer technology, which is nothing but the process of cloning. So instead of using the term cloning, they have used somatic cell nuclear transfer. And just see this. If you just break down the entire thing, somatic cell nuclear transfer, meaning that the nucleus from the somatic cell is getting transferred. So this is what this is cloning. So that's the correct answer will be reproductive cloning in animals. Now, coming to the next topic, and again, a topic which is extremely important, and this is about stem cells. Now, when it comes to stem cells, understand what we mean by stem cells. Uh, Stem cells, you can say in a manner, are the factories of the cells. These are where all the raw material gets converted into finished products. Before any cell starts to function as a normal cell, it is all a stem cell. Now, for example, we have blood cells, we have nerve cells, we have muscle cells, we have skin cells. There are n number of different types of cells, correct, in the body. And every cell has a different function, every cell has a different shape, and they are running the body, correct. They would make up the tissues will make up the organs will make up the organ systems, and the set of organ systems will make up the entire organism, as we had discussed even yesterday. So when it comes to any cell which is a proper functional cell, all these cells are born from stem cells. The raw material for all these cells are stem cells. This is where they are stemming from. In a manner, you can say that this is where they are coming from. So stem cells will be the origin for any cell, correct. So in simple terms, stem cells are nothing but the origin of every cell. Origin of every cell in the body. Now, in case of stem cells, what we see is that, okay, this is okay, fine, that this is the origin, that they are the origin for every other cell that we have in the body. But then, why do we talk about stem cells so much? Why are we talking about stem cell therapy and the fact that we'll be able to completely change some of the things that we have never heard of? So, why exactly do we think that this is going to be very, very important? And why, I mean, not going to be, but rather in the last 1 decade, we have seen that stem cell therapy has become slowly and steadily has started taking prominence. Now, why stem cells? So, one thing that we understand is that, okay, fine, they are the origin of every cell in the body. Now, overall, when it comes to stem cells, that's why, as you can see, that the stem cells can grow into all these different specialized cells. Now, with stem cells, one thing that is very, very important is to understand that they are nothing but the raw material for the other body cells, and they are considered raw material because all the other body cells specialize, all the other cells with specialized functions, they are generally formed from these cells. Now, the idea behind stem cells is, and why stem cells have become such a research topic and they have taken so much of prominence in the biotechnology area is because of the fact that stem cells have the ability to become different types of cells, meaning that the same stem cell can become a muscle cell, the same stem cell can become a nerve cell, the same stem cell can become a skin cell. So the same stem cell has an ability to transform into or grow into anything. All right. So they can actually become anything that they want. So overall, especially when it comes to the stem cells, this is where their value has to be understood, that they can just grow into any kind of cell. They can grow into muscle cells, they can grow into blood cells, red blood cells or white blood cells. They can just grow into any cell in the body. So that is where the stem cells have importance, that they have two important properties. One, their property to specialize, that they can specialize into anything, they can specialize into different types of cells. And second, their ability to renew themselves, meaning that before they start to specialize, there will be multiple cycles of cell division. And because of that, what happens is that there will be a lot of stem cells available for anything that we want to do. So these two things become very, very crucial, because these are, stem cells are unspecialized, they do not have any function, they do not have any special purpose in the body. But once they start to grow, once they start to develop, this is where they can just develop into any kind of cell and hence become useful for us. Now how they will become useful, that also we'll discuss here. So that's why stem cells have to be seen in very important light. Now, two important properties as we discussed, that they can renew themselves and their ability to specialize. So renewing themselves means that they undergo numerous cycles of cell division while maintaining the undifferentiated state, meaning that before they start to specialize, there will be multiple cycles of cell division, from 1 to 2, 2 to 4, 4 to 8, 8 to 16. We will just keep getting stem cells, right? And after that, ultimately these stem cells will start to grow, they'll start to develop or specialize into some kind of cell. So this is where their ability to specialize also becomes crucial, because they have capacity to differentiate into specialized cell types. Now, the same stem cell, so just remember that the same stem cell can specialize into various different types of functional cells in the body. Now, why these two properties become important? We'll understand that. But before that, also understand that there are different types of stem cells. There are largely three types of stem cells based on what their specialization is and where their origin is, called as totipotent, pluripotent, and multipotent cells. Now, totipotent, in a manner, this word is coming from the word total, mainly because they can specialize into all cell types in body with the addition of extra embryonic or placental cells as well. Now, totipotent cells, you can say, are the most versatile. In a manner, we can say that these are the most versatile, meaning that they have the capacity to just create anything, just change into or specialize into anything. No other cell has this capacity, or no other stem cell has this capacity, that they can just specialize into anything. So that's why all cell types in a body with addition of extra embryonic or placental cells. Now, overall, when it comes to the totipotent cells, they are embryonic in nature. Embryonic meaning that they can be derived from embryos. All right, they are derived from embryos. So that's why we can also call it as, they can be called as embryonic stem cells. They can be also called as, that's why, embryonic stem cells. So overall, what we see is that these stem cells, when it comes to totipotent cells, we are deriving it from embryos. So from the embryos, and that too within the first two cell division cycles. Within the first two cell division cycles after fertilization is where we can get the totipotent cells. So that's why when it comes to totipotent cells, it becomes extremely important to understand that they are the ones which have the most capacity to specialize. They can specialize into any kind of cell, and even not only body cells, but also they can also convert into or specialize into placental cells or extra embryonic cells as well. So they have the best capacity to specialize into any cell type, but only found in the body after the first two cycles of cell division, just after fertilization. As soon as fertilization has happened and cell division has started, the very first two cycles of cell division is where we have the totipotent cells. Then second one is a pluripotent cell. Now in case of totipotent cells, we understood that they can just convert into or specialize into any kind of cell. In case of pluripotent cells, it, in a manner, you can say that they can specialize into almost every cell. So they can specialize into all the cell types that make up the body, but not into placental or the extra embryonic cells. So they cannot really specialize into extra embryonic cells, which is a capacity that only totipotent cells have. So so here you can write less versatile than totipotent, but they are also derived from embryos. So that's why they can also be called as embryonic stem cells. So both of them are embryonic stem cells, just that totipotent in the first two cycles after fertilization, and pluripotent after the first two cycles of cell division. All right, so that's the basic difference between the two, and that's why totipotent is the most versatile and pluripotent is less versatile than totipotent. There's a spelling mistake. Hmm. So this is where we see that we have totipotent and pluripotent cells. Then after that comes multipotent cells. Now multipotent cells, with them what we see is that their ability to specialize is more limited than the pluripotent or the totipotent cells. So that's why in a manner you can say that these are the least versatile. They are the least versatile, and they are derived from the body after or during birth. So that's why they are also called as adult stem cells. So what we see is that when it comes to multipotent cells or multipotent stem cells are found in people who are born just after the birth, or maybe years after birth, or maybe in every one of us, for example. So everyone who's born, or maybe during the birth, because during the birth you have something called as placental cells, and there also you can derive the stem cells, but these stem cells will have a limited ability to specialize. So whether during the birth or after the birth, in the entire life of that individual, the stem cells that will be derived, these stem cells will have limited capacity to specialize. Now understand why they have a limited capacity to specialize. The reason for that is that these are stem cells which are found in people who have now developed. So for example, during the birth or after birth, The child already has developed all the tissues, all the organs, or the organ systems, etc. All these things have already developed, correct? So because all these things have already developed, it simply means that now uh, they can uh, all these stem cells are found in various different locations in the body now. So that's why they would have a limited ability, that if they are now located near the heart, then they can convert into any cell which is near that region. So maybe heart muscles, maybe other nerve cells and everything that you have in that vicinity, in that area. But they will not develop into a brain cell. Those stem cells will not convert into a brain cell. So now their ability to specialize becomes limited, because now the body has grown. So that's why when it comes to multipotent cells, we see that this is where you have limited ability to specialize, as compared to what we saw with pluripotent or uh, totipotent. So both totipotent and pluripotent, they are being derived from the embryos, whereas in case of the multipotent cells, they are being derived after birth or during birth. So then in that case, that's why their ability to specialize becomes more limited than others. So these are the three types of stem cells. Now, looking at how we can make the use of stem cells and where do we find uh, the stem cells being useful for us. So first is regenerative medicine. Now what we know is that stem cells can specialize into anything. Stem cells can specialize into various different types of cells. So overall, when it comes to stem cells in that sense, they can be used for something called as regenerative medicine. Now, when we say regenerative medicine, it becomes it is where stem cells can be used to regenerate damaged tissues or organs. So even for example, let's say in times of even accidents, or let's say in the times of some disease or even heart attack, during heart attack the muscles near the heart can get damaged, and these damaged muscles can be replaced with stem cells which can grow into the muscle cells of that area. So because they have the ability to grow, they have the ability to specialize, so hence if stem cells are placed in these kind of areas, they will be able to convert into muscle cells. So that's why what happens is that stem cells will be used to regenerate. It will be useful for regenerating damaged organs and tissues. Then it can be used for drug testing, where any new drug or any new vaccine that is about to be released, the first test can be done on tissues and organs which are grown out of stem cells artificially. So artificially we would try to grow tissues and organs out of stem cells, and we would do the testing there. We would look for what's happening to the organ, or is it showing some kind of an issue or any side effect, or do we see everything going fine, and is it ready to be sent to the next stage where uh, trials can start. So all these things we see that we can actually do it. So they can be used to create tissues that mimic the human organs. So this is where the researchers can use it for also understanding of the drug or the vaccine. Then it can be used in disease modeling. Now disease modeling is where we are making the use of stem cells in understanding of a disease. Now just understand this, that many a times the diseases that are developing in, let's say, a certain tissue or a certain organ, that can be understood also by studying their growth, because whether a cell is growing or a tissue is growing, they would all have grown from stem cells. So while they grow from stem cells into a specialized cell, where is that problem coming? Where do we see the tissue or the cell developing a disease? Or where is, let's say, what is the step where a certain pathogen is attacking a cell? So all these things can be understood better with the help of stem cells, and this is where we see that active research on stem cells today focuses upon, to understand the diseases, to understand what needs to be done so that that disease does not affect the body. Then I'm very sure that you all have heard about the stem cell therapy, where we have found that there are various different diseases that can be treated with the help of stem cells, where they can be useful in whether it is damaged cornea or leukemia or lymphoma, all these kinds of diseases. We see that even in blood diseases, immune disorders, in all these cases stem cells become useful, and that's why they can be used in all these cases. And then they can be useful for basic research overall, the fundamental studies of the biological processes, understanding of the growth of cells and the various aspects of the cellular growth. So all these things can be understood better with the help of stem cells. So that's why what we see is that overall stem cells become very crucial uh, in various different ways. And it's not about only stem cell therapy, but we find that they do become important for even disease modeling, better understanding of the diseases, in even drug testing, in various different types of embryonic research. So there are a lot of things where stem cells can become useful, and all that because they have an ability to specialize. Then we have a very active branch which is called as tissue engineering. So in case of tissue engineering, we see that we try to create new tissues or try to create new organs out of tissue engineering. And this is where we see that even in case of accidents or any mishaps, there also tissue engineering can be used to replace the damaged organs. So we see that there are all these things happening, and that's why it becomes a very crucial part of tissue engineering as well. And then it can be helpful for in cases of neurodegenerative diseases. So neurodegenerative diseases such as Parkinson's or Alzheimer's, etc., this is where stem cells can be useful because they can create new neurons which can replace the ones which are defective, which are diseased. So that's why in all these cases we see that they can become very important. So that's why we see that there are a lot of things that can be done with the help of stem cells, and they can be useful in a lot of different ways. Even for example, if you consider autoimmune diseases, they can be useful in fighting against autoimmune diseases as well. So in autoimmune diseases also, in dental applications, we see that they see their use. So there are a lot of things that can be done with the help of stem cells. Now, to the use of stem cells, although we do see that there are certain restrictions or certain limitations that we should be also uh, we should also understand. There are two major concerns that we need to understand. One, ethical issues in terms of stem cells, and the problems with with the adult stem cells. So ethical issues, when we talk about ethical issues in stem cells, what we see is that overall, uh, this is about the use of the embryonic stem cells, because what we understood is that embryonic stem cells are where you would be using embryos, and this is where we would be making the use of embryos to generate or harvest stem cells. And now this is something where we see that we have an ethical concern here, that you are just using the embryos for your research, in the process also killing a lot of these embryos, which is not good, because in a manner you are ending a life. In a manner, you can say you're killing a potential life by doing that. So that's why there are ethical concerns raised, that if, let's say, uh, stem cell research in embryos is just allowed without any restrictions, without any limitations, then it might also lead to commercialization of all these aspects, which is not going to be good. So that's why we need to understand that this is where ethical issues come from. And then the use of adult stem cell has a limitation, that it can only specialize into a limited number of cells. So embryonic stem cells would have been ideal to use, but we do not want to use embryonic stem cells because of ethical concerns, and we are bound to use only the adult stem cells. But the problem with adult stem cells is that they are having a limited ability to specialize. Now, uh, a method that can bridge the gap between these two issues is how uh, there is something called as induced pluripotent cells. Induced pluripotent stem cells, where we convert, this is where we can convert adult stem cells into embryonic stem cells. So this is where we say that this is induced pluripotent stem cells. So that's why induced pluripotent, meaning that we are inducing the properties of being pluripotent to an adult stem cell. So this is where we are converting the adult stem cell into an embryonic stem cell. So that's why this is something that can potentially give us a solution to these two major issues that we face with stem cells in general. Look at this question. This was asked in your paper. I'm launching the poll. So 70% of you marked B, which is actually the correct answer. Let me discuss this with you. With reference to stem cells frequently in the news, which of the following statements is or are correct? First is stem cells can be derived from mammals only. Now, there is this word only, first of all, so you should read it carefully. Now, when it comes to stem cells, although we have been talking about the context of humans, but it's not that only humans or only mammals have stem cells in them. You can say that largely the higher plants and animals, they do have stem cells. So in animals, how stem cells work, you can say that very similar to what we discussed about humans. But even in plants, we do have stem cells. All the tips of the plants, whether it is the tip of the stem or tip of the leaf or tip of the flower, everywhere, wherever you have the tip or tip of the root, wherever you have a tip in the plant, there you have the stem cells, where the stem cells will grow into the cells which are in that particular area. So this is where basically they, if you have heard of meristems or meristematic tissues, they basically come out of uh, the stem cells. So that's why the stem cells will lead to the formation of these kind of areas. So overall, when it comes to the meristems or meristematic tissue, what we see is that this is where, sorry, wait, let me rewrite this. So in case of meristems or meristematic tissues, these basically are where you have the undifferentiated cells. All right, this is where you have the undifferentiated cells, and all these will be developing into the various different types of tissues and organs in the plant, and ultimately they would keep on growing further. So ultimately, what happens is that even plants, not only animals or not only mammals, but even plants have uh, stem cells. So that's why this is wrong. Stem cells can be used for screening new drugs. We discussed about this. Stem cells can be used for medical therapies. We discussed about stem cell therapy. So that's why, yes, it can be used here. So correct answer will be 2 and 3 only. Now, the next important topic for us is DNA fingerprinting. Now, in case of DNA fingerprinting, what we basically see is that here we are referring to identifying complete or partial set of genetic information of a particular individual. Now, when we say fingerprinting, what is the uniqueness of a fingerprint? I'm not talking about DNA fingerprinting. In general, when we think of fingerprint, what is the uniqueness about fingerprint? Yes, uniqueness about the fingerprint is that it is unique, correct? So what we know is that every fingerprint is unique, every fingerprint is different, and every person has a different fingerprint. Now same goes for the DNA of two different people also, that this is where we'll see that even the DNA of two people are never exactly the same. That even if these are twins, even if these are twins of the same gender, even then there will be slight differences in their genetic makeup. So that's why DNA fingerprint is a concept which is used to look into identification of the genetic information of a particular individual. Now, because of these subtle differences and also similarities, we are able to establish relations that you will be able to, by looking at two DNA samples, you can tell whether these two samples are related by blood or these are related or not, maybe between a son and a father or anyone who has the same genetic lineage. And and there are differences that can also tell you that these may not be related at all. So overall, that's why DNA fingerprint becomes very, very useful in various different applications. For example, identification of criminals, where data analysis can be done, and with the help of data analysis of the DNA sample that we might have taken from the crime scene, we can do a cross reference and we can find out who exactly could have been present at the scene of the crime, and maybe it will take us closer to the criminal. For example, if you look at this particular figure, this is the crime scene sample that has been collected, and there are three suspects, 1, 2, and 3. Can you see the similarities between one of the three subjects with the sample? Which one is it? 2, right? You can see that this is same, this is the same, this is the same. So all these three are same. Whereas in case of suspect 1, this is different, this is different. In case of suspect 3 also, this is different and this is different. So that's why only suspect 2 is somebody who was present at the crime scene, and then after that you can, I mean, the police would take it further. So overall, this is what happens. And so identification of criminals, then it can be used for paternity tests to establish relationships between child and the parents. So this is where also it can be useful. And then claim over dead body, there also, in cases of mishaps, in accidents where you have unidentifiable bodies, unfortunately in some cases, there also DNA samples can be used to identify the people. So there also we see the application of DNA fingerprint. Then also in making effective drugs. If you remember, we discussed about something called as pharmacogenomics, right? We discussed about pharmacogenomics. genomics. So pharmacogenomics is where we are talking about making effective drugs, looking at the genetic makeup of an individual. Based on the genetic makeup of the individual, we are trying to make effective drugs. So that the drug that has been made is suitable and also functional much faster than, let's say, if we did not take this into account. So that's why understanding the genetic makeup also helps us in making effective drugs. Then it can be useful in treatment of the genetic vulnerabilities as well. So especially when it comes to genetic diseases, identification of the genetic diseases can be done much in advance with the help of DNA fingerprinting. So that also is where it becomes very very crucial, in early identification and then trying to curing it in the future. And even in wildlife management, this is something that is useful, because in wildlife management, for especially for the endangered species, vulnerable species, every species that come under, let's say, comes under the threatened group, we can do their analysis, we can do their genetic analysis, and it can give us an idea about what exactly is leading to their dwindling population, what is causing the fall in their population. So is if there are environmental problems or anthropogenic issues, we can identify all these issues with the help of DNA fingerprinting. So that's why DNA fingerprinting becomes important from the perspective of all these things, in particular. But at the same time, there are a lot of problems, there are a lot of concerns that also have been raised with regards to DNA fingerprinting. That first, what we discussed about in case of human cloning also, this is also where it can lead to reference for designer babies. And especially because we would have better understanding because of DNA fingerprinting, would have better understanding of the genes, and we can understand what is a good gene versus what is a bad gene. Correct. We would identify that this is a good gene for us or that is a bad gene for us, and based on that we would always want that the child has all these genes, that the child has all the genes which are supposedly good genes, supposedly better genes. So that's why it can all happen. And there is a concept called as germline editing. Germline editing. Now, this concept can actually give birth to germline editing, and this might become a bigger issue altogether, because when it comes to germline editing, the changes that you do, the changes that you do in case of germline editing, this is where the changes that you are doing, the changes will become permanent. Permanent in the sense that it will not be a change only in an individual, but in the coming generations. It can make the changes in the coming generations, where the primary focus, that's why here is that the embryonic cell that we have, the changes that are being done, the changes are being done in the embryos itself, and in a manner that it will be able to passed on. I mean, we can pass it on from one generation to another. So that's why this becomes very very problematic, because here you need not know about the ethics of what you are doing, because this might create ethical issues altogether, that you are taking a decision for the entire upcoming generation. You're not trying to just solve a problem, or you're not just trying to solve a disease. Let's say you're not just trying to cure a disease, a genetic disease, but where the changes that you have in germline editing, the changes that you do here, the changes are heritable. The changes are heritable, meaning that the changes can be passed on from generation to generation. So once germline editing has been done, once this kind of activity has been done, then in that case it becomes permanent. And today it's not even science fiction anymore, it is something which is a reality, that germline editing today is possible. Now if let's say germline editing is being done to solve a problem, let's say to cure a genetic disease, if germline editing is done so that in the upcoming generations that genetic disease can be taken care of, then it's okay, then it is something that is fine. But the problem is that it can go one step further, that everybody would want that, okay fine, I want my child to be something like this, that for example I want my child to be the greatest scientist in the world. I'll have to look for the greatest scientist of the current time, somehow get hold of the genes, and especially the genes which are responsible for that person being the greatest scientist of this generation, and getting hold of this gene and making sure that the child has that gene, while and inserting it in the embryos, making it a part of the embryos. So that is where once these changes have been done, the change will be permanent, and the child might grow up to become a scientist. So you do not know whether or not somebody wants to do this, but uh, it's doable today, that all these things actually become possible with regards to DNA fingerprinting. Then the genetic privacy of the individuals will be violated, and this is where again a kind of, you can say, a divide that it can bring in the society, where people might start identifying with good genes and bad genes, that okay these are the people who are superior because these are the people who have all these good genes, and these are people who are inferior because they are the ones who have bad genes. And as humans we always have liked differentiating, and also always have liked and we always find reasons to look down upon somebody else. So and from early days of the human development you'll find that we have found ways, and this would become a new way in which the society would be divided on the basis of genes, on the basis of supposedly what's a good gene versus what's a bad gene. Then the genetic information might be also misused for commercial purposes. For example, medical institutions might start telling you that okay look, you have these all genes in your body, and this particular gene can lead to this disease in the future, so you should take this medical package and that medical package. So in this manner medical packages can be sold to you, and working on your vulnerabilities and all your issues and all the fears that you have, they will build upon those fears, and they might start just pitching for all these things and selling you a lot of these things. And unfortunately this is not only about the concern over DNA fingerprinting, or the concern that it cannot be, or it might be misused. Something in general that you would see as a behavior in the society, and especially when it comes to science, that because we understand science lesser, and many a times science that's why can be misused against us. You look at a lot of advertisements, especially the advertisements of the cosmetic products or even electronic products, and how they would just advertise everything out there. And because we do not understand, we just think that okay fine, the way they are speaking seems very legit, and maybe this is why it should be fine, it should be good. So uh, let's say, I mean, take cosmetics for example, and today you have so many products which are selling charcoal. Your toothpaste has charcoal, your face wash has charcoal, your soap has charcoal, so everything has charcoal. Now why, when it comes to charcoal, is it that charcoal is so important that everybody should keep on using charcoal with whatever they can, and should we start using charcoal? Is that the case? It's not. So but they would sell you in a manner that it seems wonderful and you might want to go for it. Similarly, and every time they would work on your vulnerabilities, they would talk about that we have this formulation which will make you fair, which will make your skin white, and you fall for it. So all the fair and lovely kind of products. So all these things happen, and that is where the science is being sold to us, because we do not understand that science. We do not understand what it is, and hence it can be used against us. So whatever explanations are given to you, you just buy those explanations and uh, everything associated with it. There is even a book that has been written on this. There is a book called as Bad Science, and this book is all about only this, that how commercially science is being exploited, how you are not being told the truth all the time, and you are being just advertised with science. And there is even an instance that the author talks about, where there was a company that came to the author and they said that, see, we are going to publish a fake research, and we just want to use your name there, that according to a research under this scientist, this has been found. We are just going to just publish this research. You just give your consent that you are okay with uh, we using that research, or we publishing this in your name. He felt that this was bad, this was unethical, so he did not do it. He refused that offer. Then he says that 1 month later, he was just flipping through the newspaper and he finds that there is this article that talks about a particular research. And he then identifies that this is the same research that they had come to him with. And now what has happened is that there was another scientist who accepted this offer, and a lot of money was paid to that scientist, and the research was published. So now this was a fake research. Now, based on this fake research, a couple of months later, this company launches a product, and while launching the product, it references that article. It references those articles and those researches, that look, now we have seen that this problem and that problem happens, so you must take this product, and this product will solve your problem, in that sense. So this is how they created an entire cycle of uh, advertising, and they just published a fake research, and a product was their entire product was based on that fake research. I mean, not based on that fake research, but rather you can say that they published the fake research because they wanted to uh, launch their product in the market. So that's why we know that for commercial purposes, all these things can actually be misused. Then genetic information may be used to even create weapons of mass destruction. And this is also where we can say that genetics can be used, that once we understand genetic vulnerability, we can actually try and attack that genetic vulnerability as well. So that's why this is also something which is possible. And whether we do not know whether the COVID-19 virus was made in the lab or not. There have been reports that say it was made in the lab. China never will accept it. Now you can make them accept it. So whether it was natural or it was made in the lab, we would never know. But we have seen the kind of destruction that it caused, because our immune system had no answers. Our immune system did not know how to fight against this particular virus. And this is how you can create a weapon of mass destruction. It's not impossible in today's world. So that's why this is something which becomes extremely problematic for us, because now biotechnology has grown to a level where creating all these things in the lab is possible. And at the same time, even the pathogens which are extinct, regrowing those pathogens is also a possibility in today's world. So that's why these are all the problems that we need to understand and we need to be concerned about. Then we have gene therapy, and something that we already discussed also, that gene therapy is where we are trying to manipulate genes in one way or the other to treat or prevent a disease. Now, in this particular scenario, what we see is that either it could be about replacing a gene responsible for disease, or it could be about inactivating or removing a gene which is functioning improperly, or introducing a new gene. So, something that we discussed yesterday, that it could be about addition, removal, or modification. Modification would mean either making the change or replacing. So that's why there are all these ways in which gene therapy can be used. Now, in case of gene therapy, what we see is that today it has started becoming options for a lot of untreatable diseases. That in especially in case of cancer research, we have seen that gene therapy plays a big role there. And at the same time, there are other diseases also where gene therapy becomes crucial. Now, with regards to gene therapy, although we have seen a good growth with gene therapy over the past years, we also still see that there are concerns with regards to gene therapy. And there are three major concerns that gene therapy faces, because of which it has still not become the kind of center stage that it can take. Now, why is that? First is because of the short lived nature of the treatment. Short lived nature of the treatment. Now, overall, when it comes to short-lived nature of the treatment, understand that we are talking about gene therapy being inserted, or where, let's say, a new gene is being inserted in our body. Now, when a new gene is being inserted, in that particular sense, a new gene many a times will not be accepted by the body for very long, because in our body there is uh, a cycle of cell division that keeps on happening continuously, correct? So every second, cell division in our body keeps on happening, and every cell in the body will be born, will grow, and then will die, and it will be replaced with a new cell. And this is an ongoing process all the time in the body. For example, by the time I finish one sentence, more than 10 lakh cells in my body would have been replaced. So this is the fast growing nature of the cell and how they function. Now, in this given scenario, what happens is that uh, sometimes the gene that has been inserted, which has been introduced as a part of gene therapy, may not be accepted by the body for very long. And because of this continuous cycle of growth and development of the cell and replacement with new cells, the body might go back to the original way of working and forget about the new genes, and the new genes may not be able to translate for very long in the body. So that's why people who are going through gene therapy might have to go through uh, multiple iterations of gene therapy. Only going through gene therapy once may not be enough because of this. So that is one first concern that we have, that there's a short lived nature of the treatment. Then the immune response. Now, when it comes to our immune system has a very basic way of working, that anything that is foreign is an enemy. Anything that is foreign is an enemy. So if a foreign gene is introduced, although this gene is important, this gene is going to be helpful, immune system does not care. Immune system, anything that is foreign is an enemy. So there are chances that the new genes which are being introduced as a part of gene therapy might be attacked by the immune system, and the immune system itself may not let gene therapy work. So that also many a times becomes a big challenge in this entire scenario. Now, apart from this, one more problem has been that there are a lot of diseases which are multi-gene disorders: heart disease, high blood pressure, Alzheimer's, arthritis, diabetes. All these are cases where it's not one gene that gets affected, but there are multiple genes getting affected. So that's why treating with gene therapy becomes extremely complex in this scenario, because otherwise it becomes otherwise if it was one gene, it becomes easier for us to think of treating this disease. But with multiple gene getting diseased at the same time, uh, replacement of, or let's say uh, any modification in multiple genes at the same time is not going to be an easy task. And hence we see that in case of multi-gene disorders, gene therapy do not really work very well. Apart from all these, one more problem also in general you can say is that the technique still remains risky, that we have not really seen it to be very safe in and effective in a lot of ways. Only in areas where we see that uh, there is no other way out, there is no other therapy, there is no medical procedure, then gene therapy is used more. In some of the simpler cases, gene therapy is used more, but in very complex cases, still we see that it's not a big choice. And at the same time, cost of gene therapy also becomes a big factor, that it still remains expensive. So that also remains a problem, that because of the fact that it is expensive, it's not really possible for everyone to even go through the treatments. So all these are also issues that we have. Now, what we also have seen is that health ministry, along with AIMS Delhi, they have now started working towards various ideas that can bring down the cost of gene therapy and also of stem cell therapy, because these are therapies otherwise which are very costly, and that's why it is out of the reach of a lot of people. So that's why they are now also working on different areas where they can start making all these treatments cheaper than they are currently. So that's why there are all these concerns that we have with regards to DNA fingerprinting. So any question until now? Anything related to whatever we have discussed? So you're saying that no one has the same DNA, then how could we know about relation between parents and children? See, the entire set of DNA is not the same, but there are a lot of things which are similar. So, for example, let's say right from the analysis of the blood group of the people involved, or there are various proteins and enzymes which show similarity in case of blood relations. So that's why we look for all these clues, and if let's say all these are seen to be similar, then paternity or maternity, for that matter, can be established. What is metagenome sequencing technology? Now see, when it comes to any sequencing technology, what we understand is that overall metagenome sequencing. Now when it comes to meta, so meta means that you are basically looking towards the entire set of genes, all right, entire set of genes from all the individuals, whether it is microorganisms or humans or plants or any other organism for that matter. So sequencing of all the genes that we are trying to do for everyone, that is something that can be called as metagenome sequencing. So overall there are various researches right now which are going on with regards to metagenome sequencing, because we see that they can help us in identification of a lot of microbial mass and how exactly these microbes function. So identification overall of them can also be done with the help of metagenome sequencing. So that's why we see that it is something that is being done a lot. I mean, a lot of research is happening in this particular scenario. IVF technique can you say about it, sir? So IVF basically is in vitro fertilization, where you are talking about a bunch of different techniques which are related to reproduction, which are related to human reproduction. Where if let's say natural production is not happening, or if there is some kind of issue that the parents are facing, then in that case, this is where in vitro fertilization becomes an option. So there are a lot of ways in which in vitro fertilization is done, out of which one of the ways is what we discussed in surrogacy. So when in case of surrogacy, what we see is that the entire embryonic development will not happen in the womb of the original mother, but rather a surrogate mother. So in that case, there are a lot of these kind of procedures that are used as a part of it. Difference between gene therapy and stem cell therapy and DNA fingerprinting, these are completely different. These are three different topics that we discussed. You find differences between something which you need to find difference between DNA and RNA, because they are very close to one another in nature. But these are three different things. How to differentiate between them? Because in case of gene therapy, what we know is that we use a gene. You take a gene, and you either you replace an existing gene, or you add a new gene, or maybe you just remove the gene. So that's gene therapy. In case of stem cells are nothing but raw material for any cell type. So there is no similarity itself between the two. So uh, differentiating is not the correct question even to ask. What is placental cells? So placental cells basically are the cells which are found during the birth or just before the birth, and something that is helping in the birth of a child. So that is what we mean by placental cells. Multi-gene disorders basically are disorders where not one gene, but rather multiple genes are getting affected in a particular disease. So then we say that it is a multi-gene disorder. Haploid and Haploid and diploid, basically when we are talking about chromosomes. Haploid is when you have only one set of chromosomes. So generally when it comes to, let's say, asexual reproduction, there the organisms are haploid in nature. They have one set of chromosomes. In case of organisms that reproduce via sexual nature, then in that case they are diploid, meaning that they have two sets of chromosomes. They have a pair of chromosomes. So that's the basic idea of haploid and diploid. Recently, sickle cell gene therapy in news. So, which information is important for our examination? Only the basics about what exactly the disease is, and if we are using gene therapy, what is the basic nature of the gene therapy that is being used. That's it. Where are stem cells derived from? Any part of the cell, largely from bone marrow. Largely from bone marrow, but even fat cells in the body, they are storehouses of stem cells. Stem cell only present in the bone marrow, not necessarily, as I just said, that even fat cells is where they have. Totipotent cells. So totipotent cells are where you have um, stem cells which have an ability to specialize into any kind of cell in the body, and they are the ones which are found just after fertilization, just within the first two cycles of cell division after fertilization. For specific designer baby, is gene therapy used, or DNA should change? It's not about changing the DNA, but rather we are modifying it. We are actually uh, adding a new gene, which we feel that it is going to be good, or it is going to be helpful. So it is addition. So it is a kind of gene therapy, you can say that. Extra embryonic cells. So extra embryonic cells basically are the cells that support during the embryonic development. The tissues that support during the embryonic development, they are the ones which are called as extra embryonic cells. So placenta, for example, is an extra embryonic cell. So during the time of the embryonic development, the tissues or the cells that help or which provide an extra, uh, you can say, structure. Audio and video of the class is not working. No, it's not the case, because I'm getting the questions from everyone. Please refresh your session. Maybe it's a problem on your end. On ritual ceremony after my grandmom's death, we organized blood donation camp in which 33 people voluntarily also registered for stem cell donation for leukemia and thalassemia patients under that stem cell registry. That's wonderful. Explain wildlife management application in DNA fingerprinting. So wildlife management is where we are saying that we there are two things actually you can say that as a part of wildlife management. That one, we are trying to identify the genes and see if these genes are vulnerable to some kind of condition. Let's say if there is a huge population drop in one species or in some kind of species, then in that case what happens is, uh, we take those genes, we try to identify those genes and see whether there are any environmental factors or any anthropogenic factors that are leading to it. So overall we'll try to understand what's causing that decrease in population by with the help of analysis of their genes. So that's why in wildlife management it becomes useful. What is gene pool? Now in case of gene pool, you can say that it's a combination of all the genes which are present in any given species or any given even area. So if you have a large gene pool altogether, it uh, says that you have a better biodiversity and better chances of survival of the species. I get it that germline editing will reduce diversity and lead to discrimination, but don't you feel it is good too? Yeah, it is. That's what I'm saying, that how we use it is what matters. Now just because you want to create a species with, let's say, higher intelligence or higher abilities altogether, it may not always have a good uh, outcome for the society. Something which is going on naturally many a times has a better outcome compared to anything that we want to change. If let's say you are trying to solve a problem, then that's another thing. But if let's say you are trying to create higher species with regards to everything, we do not know how these species are going to function. You are right now just thinking about, okay, that person might have a higher level of intelligence than others. What's the harm? It's good. But when you, let's say, have everyone in that order, when you are looking towards everyone in that order, of course you cannot do it for everyone. You'll only be able to do it for some. And this is where you are starting to create a higher order of species which is definitely going to look down upon and going to act against everyone else. And this is going to create a new kind of, you can say, so-called race that will be very different from where we are right now. And you do not know what happens 2 generations later or 3 generations later. So that is where the fears are. It's not about making an immediate change. It's about, you cannot predict what will happen 50 years later or 20 or maybe 100 years later because of the actions of the present. So that's why these things have to be avoided from that sense. Also think of this in another way, that if everybody is better, then nobody is better. Then you have come back to where you are. What's the difference between stem cell therapy and covalent plasma therapy, which was used to treat COVID 19 patients? So, when it comes to the plasma therapy, this is where we actually pack the plasma with a lot of antibodies that will be taken from people who have been vaccinated or maybe others altogether who have better immunity. And then these antibodies will be packed in the plasma and then given to the patient. So, very different from stem cell therapy. Right, so many of the other questions are repeated questions. Tomorrow, I will not have a class for you because my weekend batch is also running. So not tomorrow, but it will be informed. I mean, whenever it is possible for me to take those leftover classes, I'll definitely let you know, let everyone know, and the team will inform everyone. So the next topic here is the concept of three parents baby. Now, in this particular concept, what we see is that this is where we are talking about a concept where we are trying to solve a problem that is related to mitochondria. Now, in case of mitochondria, always remember that mitochondria are transferred from the mother to the child, never from father to the child. Now, whatever mitochondria are present in the mother, the same mitochondria will be transferred to the child also. If the mitochondria are defective, then the same defective mitochondria will also be transferred to the child. Now, if father's mitochondria are even defective, doesn't matter, because it will not be transferred to the child, it will remain localized to that person. But in case of mitochondria being defective in mother, it means that the same will also be transferred to the child. Now, to solve for mitochondrial disease from transferring from the mother to the child, this is where the concept of three parents baby or three-person in vitro fertilization, etc., whatever you want to call it, or mitochondrial manipulation technology, there are a lot of names that are used here. Pro-nuclear transfer technology, there are all these names. So what we do is that in this particular scenario, if we identify that there are unhealthy mitochondria in the mother's egg cell, then we try to replace it with healthy mitochondria, where we'll take a donor, and from the donor egg cell, first the nucleus will be removed, just like what we were doing in cloning. Similarly, here also, first the nucleus will be removed, and for a very similar reason, that we don't want the specific characteristics of the donor to be there in the child. So that's why we first will take away the nucleus, and once we have taken away the nucleus, mother's nucleus will be inserted inside the donor cell, which donor cell that has all the healthy mitochondria. So donors from the donor cell will take the healthy mitochondria, the nucleus of the mother will be taken and inserted inside the donor cell, and then fertilization happens after that. So in this process, we have replaced the unhealthy mitochondria with the healthy ones. This is precisely what we have done. Now, in this particular scenario, when it comes to uh, this process altogether, instead of replacing the mitochondria in the mother's cell, we are taking a donor cell and we are removing the nucleus from there, and then we are inserting the nucleus of the mother's egg cell into this enucleated donor egg cell. Why are we going through this indirect process? Why not direct removal or replacement of mitochondria in the mother's cell? What you need to understand here is that when it comes to mitochondria versus nucleus, in case of nucleus, what we see is in case of nucleus, what we see is that there is only one nucleus in our cells, whereas mitochondria can be multiple in number. So if you want to replace multiple mitochondria, that's going to be a very difficult task. At the same time, removal and replacement of nucleus is already established technology as a part of cloning. So that's why we know how to do it. We understand how to do it, and it becomes easier for us to carry out a process which is a well established process already. So that is where we see that we can take the example of this process. Now, what we have seen is that initially the first baby was born last year with mitochondrial replacement therapy. So the concept of three parents baby, for the first time in last year in the month of May, we saw that the first example happened. Now, as a concept, it has been here, experiments have been done, research have been done, but now we see that it starts to take its shape. So that's why it remains an important topic. Although there is one question that has been asked already from here, In the context of recent advancements in human reproductive technology, pro nuclear transfer is used for. Now, it could have become a difficult question because pro nuclear transfer is not the most common name that we use for this process, but this pro nuclear as a concept, it was used in the newspapers, that it is something which is related to the same concept of three parents baby, that is prevention of mitochondrial disease in the offsprings. So that is what pro nuclear transfer is, and that's why I've not given you the poll, because it's completely new term that we have not discussed yet. Then, with regards to GM mustard, now when it comes to GM mustard, we have seen that GM mustard constantly has been in the news. This GM mustard is called as Dhara Mustard Hybrid 11, or in short called as DMH 11. Now this DMH 11 is a hybrid. All right, it is a hybrid, and this is where we see that we are talking about a hybrid which is going to be used in India. Now there has been a lot of controversy around it. There has been a lot of controversy around it, that it has been in research for a very long time. I think since 2004-5 they had started working on it. 2011 onwards field trials had started, and we saw that it can give up to 25 to 30% better yield as compared to the even the best varieties of mustard that we grow in India currently. So that's why from that perspective we see that it looked good, and a lot of field trials have been done, a lot of experiments and research have been done, and ultimately it was ready. It was said to be ready. It was prepared at the genetic center for genetic manipulation of crop plants, which is present in Delhi University. Now in this particular scenario, what we are looking at is a better productivity. So it looked good for the market, and it was about to be released for the Indian markets back in 2018, and it was also cleared by the Genetic Engineering Appraisal Committee back then, and the government was also ready to launch it for the Indian farmers. But this is when Supreme Court intervened, and Supreme Court put a stay on the release of these seeds. And then Supreme Court said that we need to study it further and then come back and look at all the possibilities, because there were a lot of concerns that were raised against this DMH 11. So because of which ultimately we saw that it was not released back then in 2018. But then we see that last year again it comes back in the news in the month of September and again in the month of November, that first we were talking about uh, GM mustard and how GM mustard was about to be released. Because what happened back in 2018 was that the Supreme Court said that a committee should be set up, and this committee should study everything with regards to GM mustard, understand if there are any issues that they see, if they come across. If not, good. If they come across, whatever it is. So they have to just submit their report whenever they are done. Now, the committee finally in 2022 came back with their report, and they submitted the report, and they said that okay, everything is fine, we do not see any problem with this GM mustard. And again, Genetic Engineering Appraisal Committee has again cleared it for the release. Now the center, the government is seeking permission from the Supreme Court to go ahead. So we see that this is where the case is still pending, and the Supreme Court still hasn't said anything with regards to whether it can be released or not. So this is where we are right now. That again, the matter has been given back, has been sent back to the Supreme Court, because in November also we saw that the government was pushing very hard and was trying to get a date from the Supreme Court so that they can move forward with the matter. And ultimately, this news came in November, that it already seems to be very late, because the sowing season for mustard is maximum December first week, but we already are in November second week, and we do not see anything happening until now. So it seems like it may not happen. That it for this year the release may not happen, and again it gets postponed to the next year. So overall, when it comes to GM mustard, there are a lot of pros and cons that people have been talking about, that what is important, what can be important, and what is wrong. So if you think of the positives, in case of the positives that we see with GM mustard, with regards to GM mustard, the positive that we have is, first and foremost, when you look at mustard consumption in India, mustard consumption in the entire country is pretty high, right? And in one way or the other, mustard is being consumed, whether as mustard oil or whether as mustard seeds, but mustard consumption is huge. Now, a lot of mustard in India is imported. We do not grow enough of mustard, and majority or more than 50% of mustard is imported. So that's why uh, the cost also increases, because you are importing mustard in a lot of cases. So obviously cost is higher, and productivity of the indigenous mustard variants is not that great. So that is where this GM mustard can help. It will help in bringing up the productivity. So you have better availability of mustard in the domestic market, so prices of mustard might fall down, prices of mustard oil might fall down. At the same time, for the farmers also it could be better, because farmers will be able to get better productivity or better yield out of this GM mustard. So that's why it might also help in increasing the farm income. It is also seen in many of the articles, or many of the many of the points that have been put forward by many experts. What they say is that this could be equivalent of what happened with BT cotton, that as we moved from cotton to BT cotton, the farmers also gained a lot. The farmers could also get a lot of benefits out of it, and it helped in increasing their farm income. So something similar might happen with GM mustard also, that it might help the farmers increase their farm income. So that's why these are all the positive points which are kept in the favor of GM mustard. But at the same time, there are points that go against it. One problem has been from the perspective of the safety of the food. That are we completely sure that it is not going to be unhealthy? It is not going to cause some problem in the long term? Or are we just looking at a short term analysis and saying that everything is fine? Is it something that can cause problems 5 years later or 10 years later, and by that time it will be very late for us to act on it? Something what has happened with some fertilizers and the chemicals that are used in those fertilizers, the toxins that are used in those fertilizers and pesticides. So can something of the similar nature can happen here? So there are these concerns raised, plus there are environmental concerns from the perspective of biodiversity. Right now, there are 66 variants of mustard being grown in India. And once GM mustard becomes a reality, obviously a lot of these mustard variants will not be grown anymore, because everything will be replaced with this GM mustard. Because GM mustard gives you better uh, productivity, better yield. So why not move to it? Farmers might stop cultivating the older variants of mustard and move to GM mustard now. And this might lead to decrease in the genetic diversity of mustard, which is always bad. Biodiversity has to be maintained, whether biologically, whether genetically, whether species level, but always biodiversity is good for the survival of any particular species. But here what's happening is that you might decrease the survival chances because of decrease in biodiversity of mustard. So that's why there are these points which are being raised against GM mustard. So that's why there are all these problems that we are facing. This article, I would say, is a wonderful article to read. I mean, you just go and Google it if you can, and find this article in the Hindu. It's a wonderful article to read, and a lot of points with regards to GM mustard. This question was asked in a 2019 paper. Read the question, please. I'm launching the poll for you. I'll just give you a hint that there is one statement that makes this question very, very easy. Hmm. So 58% of you have marked the correct answer, which is B. Let me discuss this with you. Forget about statement number 1 and 2. Let me just directly bring you to statement number 3. GM mustard has been developed jointly by IARI and Punjab Agricultural University. We have just seen that it has been developed where? It has been developed at Delhi University. So this is wrong. Correct answer is 2 only. So first two statements we haven't even discussed, and it's not even required to answer this question. First statement says GM mustard has the genes of a soil bacterium that gives the plant the property of pest resistance to a wide variety of pests. This is largely about BT or the BT variants in general. We see that this is where soil bacterium are used, but in case of GM mustard, that is not the case. So that's why this is wrong. GM mustard has genes that allow plant cross pollination and hybridization. Uh, this is correct, and this was also raised as a concern that because it has a possibility of cross pollination and hybridization, the genes might also be transferred to some other variants, and it might cause bigger challenges there as well. So we need to understand all those problems. But only statement number 3 is enough to answer this question, because as we discussed here, that it was developed by the Center for Genetic Manipulation of Crop Plants, which is located in Delhi University. Let's take a break here. We'll start with dark biotechnology when we come back. Let's be back by 7:55. It's 7:42 right now. Let's be back by 7:55, and let me take things further. This is the last topic of discussion for this chapter, and then we'll move on to our next chapter, that is nanotechnology. Welcome back, everyone. Now we are here in the last topic of discussion in biotechnology chapter, and this is dark biotechnology. Now, in case of dark biotechnology, it is about misuse of biotechnology. Now, in all these topics that we have been discussing until now, we also discussed that it's not only all the advantages that we have, but there are certain limitations. And not only limitations, but there are aspects where it can completely take a negative turn. And when that happens, this is what dark biotechnology is about. So whether it is about creating weapons of mass destruction, whether it is about bioterrorism or bio warfare, in all these cases we can say that this is an aspect of dark biotechnology. Now when we say, for example, that's why bioterrorism, bio warfare, biocrimes, anti-crop warfare, in all these cases we can call it as a part of negative aspects of biotechnology, and hence dark biotechnology. Now that's why, in simple terms, you have to understand that we are talking about misuse of biotechnology. Misuse of biotechnology. So misuse of biotechnology, or all the negative aspects of biotechnology, can be called as dark biotechnology. Now there are again multiple examples here. First, looking at bioterrorism. Now bioterrorism, when the word terrorism comes, it's not only about harming a human life or only killing a human life rather, but it's also about anything you can do against a certain country, anything that you can do to negatively impact, let's say, India. So everything then in that case will be called as an act of terrorism, or if biological agents are used, bioterrorism. Now for example, let's say uh, somebody releases some kind of pathogen, some kind of pest that is going to negatively impact Indian agriculture, and it leads to a huge drop in the productivity of Indian agriculture. Even that will be counted as negative aspect of biotechnology or an act of bioterrorism. So that's why anything that can not only hurt us physically, not only kill populations, but rather at the same time also hurt economically, can be called as bioterrorism or an act of bioterrorism. At the same time, there are bioweapons that can be made. We discussed about the case of COVID-19, that how COVID-19, if it was created in the lab, it was an act of bioterrorism, or it was a bioweapon that was used. And it's not even the fact that it's not impossible. We know that there are certain other cases also where we have been able to do that. If you consider this regeneration of the extinct pathogens, which is a possibility now, and we have seen that happen. There was a news article last year and very recently also, even this year, this was again in the news, about scientists reviving zombie virus which spent more than 48,500 years in frozen in the Arctic. Now first of all, why would you even think of doing something like this? Because we have just come out of COVID, and it's not completely yet, but we have seen the kind of devastation that it could cause. And after that, if people are still looking to regenerate extinct pathogens or do all these activities, it's not safe. We do not know what turn it can take, so we have to be very closely watching all these things so that it does not really lead to anything against humanity. So all these are possible. Now let's say, for example, if you take the case of any disease which now does not affect us, something like a plague or smallpox, which have been nightmares for the human populations in the past, but then wide vaccinations have happened and all these activities have ensured that they do not happen anymore. Now if you consider smallpox, for that matter, smallpox has been eradicated, but pox virus remains. Smallpox virus still remains. What if tomorrow smallpox virus is replicated in large numbers and it is unleashed on the human population? The generation today does not have an immunity against smallpox. It had years when in the years when the vaccinations were happening. So now the new generation is not being vaccinated against smallpox anymore. If smallpox is unleashed in one way or the other, it could again lead to an epidemic or maybe a pandemic of this nature. So that's why we need to understand that today, because of the fact that all these are possible, it becomes very, very dangerous in this scenario. Similarly, it also has effects on biodiversity. Now, how it has effects on biodiversity, we took an example of GM mustard, and how we discussed how GM mustard brings in a scenario where it can have a negative impact on biodiversity, that it can decrease the diversity of the mustard species. But at the same time, there is a larger problem here as well. The larger problem is when you look at all these species where we are focusing, when you look at the number of species that we are focusing on, whether plant or animal species, they're just handful. I mean, not even 0.01% of the entire species pool that we have on the earth. So then in that case, the entire study, all the researches, everything that is happening is on a very small bunch. So we are ensuring that this small bunch of flora and fauna prospers, and we are giving every support possible to it, because these are the species which are so called important for us or useful for us. They have some kind of application for us, and hence we are focusing on them, and we don't care about 99.9% others. So overall, this becomes a crucial aspect of every problem that arises from here, that we are only focusing on a handful of species, and there are large number of species which we are just neglecting because they are not useful for us, right? So that is where the problem begins from, and that's why it has a wider problem associated with a loss of biodiversity and every problem that can arise out of loss of biodiversity. So that's why we have to understand all this. Similarly, there is this concept of terminator genes. Now, in case of terminator genes, we are talking about specific genes which are inserted in the seeds so that the plants germinate, the seeds germinate only once. Suppose that you have gone to the market to buy some seeds, you brought these to your home, you used it in your farm, in your agricultural field, and you had wonderful crops. You saw very good quality and very good quantity with those seeds. So you obviously would want to preserve those seeds so that in the next season again you can use the same seeds, because you have seen very good produce qualitatively and quantitatively. So that's why you would want to preserve it for the next season. Now in the next season again you use the same seeds taken from these plants, you sow them in your fields, and you see nothing. None of those seeds germinate. The seeds do not germinate because terminator genes were used in them. And if terminator genes are used, it would mean that the seeds will germinate only once. So that's why it's it stops the plant from releasing fertile seeds. All right, stops the plant from releasing fertile seeds, and hence the farmer again is required to purchase the seeds in the next cropping season. So that's why just remember that when you use terminator genes, the seeds germinate only once if terminator genes have been used. All right, so that's why we see that this is something that becomes very, very crucial for us to understand, that how it can have a negative impact on agriculture. And it's all for the commercial gains, because now you are making the farmer dependent on, the company is making the farmer dependent on that company, because the farmer saw very good produce, saw very good results, so obviously they would be bound to come back and buy the same seeds again. And by this time the seed prices would also have risen. So they are trying to capture the market by doing this also, which is not good, which is also unethical in a manner. And because they've not told, the seeds are not coming with a disclaimer that look, the seeds have terminator genes, so just beware. Obviously they're not coming with that disclaimer. So that's why it's an unethical practice. At the same time, there are larger problems that it can create. The larger problems in the sense that the same terminator genes, because of cross pollination or maybe because of hybridization, the same terminator genes might transfer to another plant, and become part of the genetic uh, variation there. So what then happens is that the same terminator genes are getting transferred to other crops which are growing in the same field. So then in that case what happens is that not only this crop suffers, but all the other crops being sown there also suffer, and that's a larger problem for the farmer then. So that's why we have to understand that this is also possible, that they may also cross pollinate with the local variants and may affect the continuity of agriculture altogether. So that's why this is also one of the ways in which negative aspect of biotechnology can be understood. So that's why just understand this, that there are a lot of ways in which it can be misused. Some of the misuses we also discussed in the terms of uh, DNA fingerprinting and the problems associated with even GM mustard. And so all these things also we understand that everywhere there is some issue that we have. So negative aspects is what we have to worry about, whether it is the concept of designer babies, whether it is a concern with regards to human cloning. All these things will be counted as a part of dark biotechnology. Now, coming to some of the questions that have come from biotechnology in the past, again, the questions have been easy in nature. They haven't really asked difficult questions. If you see the first question, 2021, what are the research and developmental achievements in applied biotechnology? How will these achievements help to uplift the poorer sections of the society? Now, when they say applied biotechnology, even if you don't know what is applied biotechnology, you can think of what it is. When you are applying biotechnology, when you are using biotechnology for various different applications, basically what the entire chapter was. So you have to just summarize everything that we discussed in this chapter. So research and developmental achievements in applied biotechnology. So you can again go sector wise, that in medical applications these are the achievements, in agriculture these are the achievements, in industries these are the things that we have done, in the environment these are the things which are done. So you can talk about all that and how these achievements help to uplift the poorer sections of the society. Now there are two aspects that you can think of. One, from medical purpose, that how cheaper medicines are made, more effective medicines are made. The medical procedures are becoming cheaper because of this. New procedures coming in, and all these procedures also they are helping in providing better healthcare support. So you can write about all that. Plus you can write about agriculture, that how it helps in agriculture, how it helps the farmer, and hence how it helps in entire bringing in, let's say, better opportunities, better productivity, and hence better income in all these places. So that's why there are all these points that you can discuss here. Then the second question: how can biotechnology help improve the living standards of farmers? Now, this is a question mainly about agricultural applications of biotechnology, correct? Nothing other than that. So, every application that we discussed, it's more about that. Just that you'll have to change your language while writing your answer. You cannot directly just list out all the applications of biotechnology in agriculture here, because the question is asking how does it improve the living standards of the farmers. So, then each and every point that we discuss, just connect it with the farmer and write from the perspective of the farmer. Although you will write the same points that we discussed in the paper, I mean in when we discussed the agriculture part. So you have to write the same points, just that in a different language. And then third question: why is there so much activity in the field of biotechnology in our country? How has this activity benefited the field of biopharma? Now, why so much activity is the first question. Why so much activity? Now, why so much activity also has a simple answer, that we see a lot of potential. We discussed about what all can be done with the help of biotechnology. So you just again have to summarize all these important points, and that's why then you have to say that that is why we have so many activities in the field of biotechnology. And then the second part is about biopharma, that how has this activity benefited the field of biopharma. So then you have to write about all the research and development in the field of medical science, and what are the various aspects of medical science that is being helpful, that whether about insulin we discussed, or effective drugs, or drugs taking care of the genetic variation, pharmacogenomics, that is how pharmacokinetics helps in bringing in better and more effective drugs. Then how better vaccines and faster vaccines are available. You can give an example of COVID-19 now, that in case of COVID-19, we saw that the vaccine was available within 6 to 9 months of time. Normally, vaccine development in the past has taken enormous amount of time. So all these points can be used in writing answer to this particular question. Then the fourth question, if you see, stem cell therapy is gaining popularity in India to treat wide variety of medical conditions, including leukemia, thalassemia, damaged cornea, and severe burns. Describe briefly what stem cell therapy is and what advantages it has over other treatments. So overall, in case of biotechnology, what we understand is that stem cell therapy is a very crucial aspect. And what is stem cell therapy, they have asked you. Now understand this question, if you see, the first part of the question is not even a question, but it's a statement about stem cell therapy. It's just a statement about stem cell therapy, where it is talking about how stem cell therapy is useful. All right, how stem cell therapy is useful, and how it can solve various problems. So it's talking in a very positive tone, correct? It is talking about very positive tone. It has not asked you about what are the problems, what are the limitations, what can't be done, right? So you shouldn't go there. It shouldn't be that you have just started listing down all the problems and all the limitations, and about this cannot happen, that cannot happen, there is an ethical issue, there is a problem, there is a money problem, it is very costly. Please don't go there, because the question has not talked about anything like that. It is not asking you to critically examine or talk about let's say the positives and the negatives, the issues. No, it just says what is stem cell therapy and what are the advantages that it has over other treatments. So that's why, because it has a very positive tone, while writing your answer also, your tone should also be positive. You should not take a negative tone here or a balanced approach here. Talk in the favor of stem cell therapy. And that's why, just remember that wherever you see such a positive vibe around a particular question, you always have to maintain that positive vibe while writing your answer as well. So that's why it becomes very crucial for us to match the tone of the question. If the question has a neutral tone, write in a neutral tone. It, let's say, sometimes, let's say it could be about uh, critically analyze or something like that, or maybe some negative aspects that they have talked about, then you have your freedom that you can use your negative points as well. And but when it comes to a question like this, where you have such a positive scenario, where everything only in terms of the positives of stem cell therapy is being talked about, you should also maintain that language, right? So any question before we move on to the next chapter? In news, there is a chip which is used to connect with brain. What is that? It was there in the news yesterday. If you go through yesterday's newspaper, it was there in the newspaper yesterday, and the Neuralink thing. So if you just go through it, it will help you, or go through the Hindu analysis of yesterday. We have discussed it in detail there. Investment diversification can be a point. For example, drought resistant technology can help save the crops, or say not using pesticides or more fertilizers in genetic modified crops can help save money. Yes, of course, this is a legit point, and of course, this is a point that you can use. Neuralink, yeah, Neuralink is article in yesterday's newspaper. Kumar, the answer to your question is no, not at all. Can I send you the answers for evaluation? Yes, of course, you can send it to me. Hybridization. So hybridization is where we create hybrid varieties of any plant, where you are maybe mixing the genes of two different crops or plants. So that is what is hybrid varieties. So GM mustard, just understand that GM mustard is nothing but a variety of mustard where we are creating a mustard variant which has better potential with regards to its uh, potential for productivity or yield. So yield is going to be much better. That's it. In the question of why so much activity around biotechnology, can we mention about success of green revolution based on hybrid crops? You can say that up to an extent, but the problem there is that in green revolution, it was not about a lot of genetic activities. There were hybrid varieties that we used. So technically, it is biotechnology, but when we uh, talk about biotechnology in general today, we are talking about genes, and wherever genes are used in one way or the other, that is where we are taking about it. So that's basically where the difference lies. So you can mention it that that was the beginning, but you cannot take it as a prime example. Hashmeet sir is actually taking another batch in the evening right now. So in this entire month, he'll be taking the evening batch of polity. If biotechnology is used in correct way, we can be superhuman in nature. Long way from there, but yes, why not? What is the difference between hybrid and gene variety? So basically, when it comes to any hybrid varieties generally is a much simpler process where genetic manipulation is not being done. That it's not that we are taking a gene and inserting recombinant DNA technology is not being used. So basic difference will be that wherever you are using recombinant DNA technology, that is where you have genetically modified crop. Otherwise, these would be hybrid varieties. All right. So one question already is, what is nanotechnology? We are just about to start it. All right. So nanotechnology, the next chapter. Now, when we look at the examination or the perspective of the examination first, nanotechnology is where we are talking about technology which is related to a very small nature, let's say a very small scale. And from examination perspective, what I would say is that it is a chapter more important for your prelims than mains. All the questions have come in mains also, but the orientation of chapter is more tilted towards your prelims examination than mains examination. Now, what is nanotechnology? So for that, again, as we did in the last chapter, just break it down into two parts: nano and technology. Now, the meaning of the word nano. Meaning of the word nano. Nano is a term that is used for anything that is very small. All right, we discussed about nano satellites because these are satellites which are small satellites, 1 kg, 10 kg in weight, and so on. So that's when nano. we have the Tata Nano car. So Tata Nano car, because they created a compact car which was a very small car compared to others, so hence they named it as Tata Nano. So nano in general is a term. I mean, in Latin it means dwarf. The term means dwarf, so that's when nano is a term that means dwarf. So anything that is very small, we use the term nano for that. So that is where it comes from. Now, in this particular scenario, when we say nanotechnology, it is the technology of the small. But how small? This is where we are going to the scale of 1 nanometer. All right, we are going to the scale of 1 nanometer. How much is 1 nanometer? How much is 1 nanometers? Yes, 10 to the power of minus 9 meters. 10 to the power of minus 9 meters. So 1 nanometers is equivalent to 10 to the power of minus 9 meters. And this is precisely the scale that we are talking about. Any material, any particle which lies in this scale, the scale of 1 nanometer, maybe 1 to 100 nanometers, they are called as nanomaterial. And the science associated with them is called as nanoscience. The technology associated with it is called as nanotechnology. Now, overall, when it comes to nanotechnology, what we understand with nanotechnology is that this is where we are talking about a scenario where these small uh, matter or small particles is what we are discussing. So overall, when it comes to this, understand why 1 nanometer. All right, the importance of this scale. Why 1 nanometer? Because there is no micro science or micro technology or milli technology and so on. We did not name any technology after any given scale. But what is so special about this scale that we named it with nano, we named it after it. The main reason why we have done it is because this is where the properties of any matter start to show up. Any property that you see at your level, they start developing first at this nanoscale. They start taking shape at this nanoscale. Before this nanoscale, if you are going beyond this nanoscale, then you have particles which have very similar nature. But when it comes to the nanoscale, this is where you have the scale of the molecule. From the atomic scale, now you have come to the molecular scale, and molecule is where you see that all that properties taking shape. Just take as an example, let's say you have hydrogen and oxygen. The atoms of oxygen and atoms of hydrogen will always have the same kind of properties. I mean, they would have their own properties. But when they combine, when hydrogen and oxygen combine, they can either make H2O, water, or maybe they can also make H2O2, which is hydrogen peroxide. Now, hydrogen, we discussed that hydrogen is a fuel, oxygen is an oxidizer that helps in combustion, and water can extinguish fire. So completely opposite properties than what the original properties of hydrogen and oxygen are. Similarly, when you look at the difference between H2O and H2O2, water and hydrogen peroxide. Hydrogen peroxide can be used as an antiseptic. It has antimicrobial properties. It can be used as an industrial reagent. It is a very good oxidizer. So there are all these properties that hydrogen peroxide has, but it is not drinkable. It is not consumable. Water, I need not tell you what are the importance of applications of water. So but we know that they are very different in their properties just because of one extra oxygen in the structure. Just one extra oxygen, and this combination has completely changed the properties of hydrogen peroxide compared to water. So that is where we see that this is where nanoscale starts. This is where the nanoscale starts, because this is where the molecules are formed. And forget about hydrogen and oxygen being two different elements combining together to form these compounds. Just think of carbon. We discussed that carbon is coal, right? Coal is nothing but carbon plus maybe some impurities. Then there is something called as graphite and even diamond. All these are nothing but carbon only, right? All these are carbon. But when it comes to this carbon, graphite and diamond are also, these are also called as allotropes of carbon. We also call them as allotropes of carbon. Now, graphite and diamond have completely different properties. Although both are made of only carbon, they have only carbon, but they have completely different properties. Diamond, you know, it's a precious stone, very costly, becomes an ornament for you. And graphite, you use in pencils. So, completely different properties. Why? Just because arrangement of carbon atoms among themselves is different. That's it. So, and all this is happening where? All this is happening at nanoscale. That's why nanotechnology. That is the importance of nanotechnology, because this is where any particle starts to adopt its properties. All the properties that you see in a material, all these properties are adopted at this particular scale, and hence this scale becomes crucially important. So that's why this term nano comes from the Latin word for dwarf, and this is where we are talking about a first point where we can assemble something, and this is where they start gaining their properties. Now, the term nanotechnology was coined by a Japanese scientist in 1974. The name was Norio Taniguchi. He was the one who had coined this term, that is nanotechnology. Now, overall, when it comes to nanotechnology, we are talking about a branch of science that deals with the minute particles in the range of 1 to 100 nanometers in size. So, we are talking about minute particles which are in the range of 1 to 100 nanometers. Now, when it comes to this particular scale of nanotechnology, this is where we see that nanotechnology becomes useful, because this is where you have the molecular scale. Although even if you, let's say, have 0.1 nanometer, does not mean that you cannot classify it as a nanomaterial. It will still be called as a nanomaterial. So everything that is in and around the range of 1 nanometer size, all right, anything in and around the range of 1 nanometer size can be called as nanomaterial. So overall, this is basically what we see. Now, when it comes to nanomaterials, we see that this is where all these properties they start to gain. All the properties are now visible, and even manipulation of that technology or manipulation of that property is even possible. Today, what we see is, or in general, what we say about nanotechnology is that we are still in a very nascent stage of development of nanotechnology. Nanotechnology, what we are doing with nanotechnology is still very early days compared to what nanotechnology is capable of. How nanotechnology can completely transform technology. Compared to that, what we have achieved is just probably the surface of what we can actually do. So, but it becomes very complex as you start going in that direction. It becomes very complex, and that's why it's not a technology that can be very easily adopted. It takes a lot of time and effort and a lot of investment for anyone to even think of adopting nanotechnology. There are, I mean, potentially there are a lot of things that we can do with nanotechnology. So if you look at the nanomaterials, this entire segment, these all can be called as nanomaterials, right? These all can be called as nanomaterial. So that's why I was saying that even 0.1 nanometer can be considered as nanomaterial. And as you see, as in comparison to that, even blood cells are bigger in size, virus is bigger in size. So that's when nanotechnology can be used even in making drugs. We'll discuss that later, that you can simply inject the drugs inside the blood cells, and with the blood cells they will be carried to the different parts of the body. So targeted drug delivery can be done with the help of nanotechnology. So that's when nanotechnology becomes extremely important in that particular scenario. And we'll discuss about carbon nanotubes also. We'll discuss about various different nanoparticles where you'll understand how they become crucial. Now, if you just look at the brief history of nanotechnology compared to other branches of science that we have discussed until now, whether space technology or various factors in energy or even biotechnology. Compared to that, nanotechnology is a fairly new science. The term nanotechnology itself did not exist until 1974. It was just coined in 1974. The first instance, though, was 1959, when a very famous scientist, Richard Feynman, was giving a lecture called as Plenty of Room at the Bottom. Plenty of room at the bottom. And this is where he had explained for the first time that how everything that we are doing right now is more associated with what we see, what we observe, but we do not know what lies beneath, because we have not even thought of what happens beneath what all we see in our life. And there was nothing, there was no instrument that could have helped us in seeing what was at the nanoscale. There was no microscope that was capable of looking at the nanoscale yet. So he said that our research now should start focusing there, on what we can't see, instead of just focusing on what we can see. And this became a very famous lecture. And Richard Feynman in general has been a personality who was well followed in the scientific community, and not only 1950s, but you'll see that until his demise, he has been one of the most followed scientists of the generation. And even the book that was published on his biography, Surely You Are Joking, Mr. Finman, became one of the best sellers. And it is very rare that a scientist's biography could become a bestseller. So, and it's a wonderful book to read. I mean, if at all you get time, it's a wonderful book to read about his life and everything that he has done, and so many adventures and so many weird things happening in the life. So, and that's why the name of the book aptly has been given, Surely You Are Joking, Mr. Finman. So he basically was the one who, he has been a big name in nanotechnology, a big name in quantum research, and hence his when he said this, it brought in a lot of following from the scientists, from the students, and everyone started focusing on this branch. They started doing research in this branch. And then we see that the next thing was the coining of the term nanotechnology. So in a manner, it gave shape to this entire segment, or entire sector of nanotechnology. But the biggest change happens in 1981, when scanning tunneling microscope is invented. Scanning tunneling microscope. Until now, until now, only theoretical research was happening. There was no practical research because we could not see at that level. There was no microscope that could see at that level. So that's why everything that we were doing until now, it was nothing but only a theoretical research. But this is where we start moving from theoretical research to a practical application with the invention of scanning tunneling microscope, because this microscope gave us the ability to look at nanomaterial, to see nanomaterial at this particular scale. So that's why this became very crucial from that perspective. The scanning tunnel microscope, they were able to, they gave us the ability to analyze nanomaterial. So that's why we see that this takes a very important turn now. Now just understand also what is scanning tunneling microscope. Now scanning tunneling microscope is based on two basic principles. One principle is the principle of tunneling, and the second principle is the principle of piezoelectric effect. Let's try to understand what these two mean. Now, when it comes to scanning tunneling microscope, typically this is what the microscope is like, that there will be a metal tip that will be incident on a surface. So there is a metal tip, and this metal tip is placed on this surface, or in a manner you can say that it applies some pressure on this surface. And as it applies pressure, we see that electrons are released, and these electrons are able to break the energy barrier and enter inside this surface. they're able to break the energy barrier and enter inside the surface. So something like this happens, that let's say you have a surface like this or a barrier like this, then an electron which is traveling, an electron traveling will be able to travel and get inside this. All right, it will be able to move inside, break the energy barrier of this surface or this substance and move inside, and hence it will be able to analyze at the nanoscale nature. So this is where we see that the wave nature of electrons is actually utilized in case of, and this is called as tunneling, because electrons are making a tunnel, right? They are making a tunnel through this surface or through this substance, and hence it is called as tunneling. Have you seen the film Ant-Man? There's a Marvel movie called as Ant-Man, and if you have seen the film, you might have seen that the characters who you have there, they shrink and they are able to enter inside surface. So that is nothing but tunneling. This is what tunneling is. So tunneling is where you are able to break that energy barrier and enter inside. Unfortunately for humans, outside movies, for humans it is not possible, but for subatomic particles like electrons or light particles, etc., for them it is possible. So this is what precisely tunneling is. So tunneling is used here, quantum mechanical effect of tunneling, that allows us to see the surface. So tunneling, in case of tunneling, what we see is that this is where electrons break the energy barrier of a substance and enter inside. Okay, so this is basically what we see with regards to uh, tunneling. So that's why in a manner they make a tunnel through this. So in, I'll just write it here, in other words, they can make a tunnel and enter the substance. So this is what happens in tunneling. Now what we are saying is that as we apply some pressure through this metal tip, electrons are released. This phenomena itself is called as piezoelectric effect. Piezoelectric effect, basically, by definition, piezoelectric effect is when electricity is produced because of application of mechanical stress or pressure. So when you apply pressure, if by applying pressure on a crystal solid or any solid, if let's say electrons are produced or electricity is generated, then it is called as piezoelectric effect. So piezoelectric effect is electricity generated, electricity generated by applying mechanical stress or pressure on a substance. Okay, so electricity generated by applying mechanical stress or pressure. So that's why piezoelectric effect works here, that when pressure is applied on this metal surface, then electrons are released, meaning that electricity is generated. So that's why piezoelectric effect. Last year piezoelectric effect was also in news, and it was in the news in the context of that until now piezoelectric effect only has been observed in solids, only in the solids. But for the first time, scientists were able to see piezoelectric effect in liquids as well, that when they applied pressure on liquids or on a particular liquid, they saw that piezoelectric effect was working there, that electrons were released, or electricity was being generated by applying it. So for the first time it has been observed in liquids, otherwise until now it has been a property of solids. So so that's why tunneling and piezoelectric effect together, these two are the quantum mechanical effects which help in uh, the working of scanning tunneling microscope. Now there's another microscope called as atomic force microscope. Now atomic force microscopes basically uh, takes care of one limitation that the scanning tunneling microscope has. The limitation is that since you are talking about electrons entering the surface, you are talking about metals, because electricity can pass through metals, electricity cannot pass through non-metals or through insulators. So you are talking about conducting materials only. So when you are trying to analyze a non-conducting material, then in many cases scanning tunneling microscope is not useful. That is where another microscope was invented later, called as the atomic force microscope. And this atomic force microscope has the capability to even analyze the non-metallic surfaces. So that's why it overcomes one limitation that scanning tunneling microscope has. So what we do here is that here also there will be a, a, a kind of a tip, and this tip will be trying to go through the surface, go over the surface and analyze the surface. So as the tip moves across the surface, it interacts with the sample's atoms and generates a three-dimensional map of the surface. And this is where we will be able to understand the surface or analyze the surface. So that is where we see that it becomes crucial, because atomic force microscope can be used for non-conducting material where scanning tunneling microscopes are not useful. All right, so this is where atomic force microscope can be used. So now, with the help of these microscopes, we were able to find or discover various different types of nanomaterial, different types of nanoparticles. Now all these nanoparticles that we are discussing here, fullerenes were the first nanoparticles to be discovered, and for the discovery of fullerenes even Nobel Prize was given. Now here on you'll find that a lot of Nobel Prizes that have been given in the last 30 odd years, they all have gone to nanotechnology, because nanotechnology has been one of the newest sciences of the last 30 odd years, and that's why a lot of new developments kept on getting new uh, Nobel Prize. I mean, every topic perhaps that we are going to discuss from now on has a Nobel Prize in its name. Scanning tunneling microscope, fullerenes discovery, carbon nanotubes, after that quantum dots, everything that we are going to discuss, you'll see that there is a Nobel Prize that has been given to it. So fullerenes were the first ones to be discovered. Now, with regards to fullerenes, we are saying that it became the third form of carbon along with graphite and diamond, meaning we are talking about another allotrope of carbon, a new allotrope of carbon. Now, in case of fullerenes, the structure of a fullerene is something like this. As you can see, it's like a football-like structure where the threads that you have, the vertices that you have, these are the carbon atoms. All these ball-like structures that you see right here, these are the carbon atoms. All right, these are the carbon atoms, and they are combined in a hexagonal way, right? They are combined in a hexagonal way. So you have a hexagonal structure which is making up a spherical structure altogether, hollow from inside, and this is how the arrangement is. The fullerenes were discovered. Now fullerene discovery was very important, and this kind of fullerene is also called as Buckminster fullerene. There are different types of fullerenes. You have C60, C72, C75, etc., meaning that there are 60, 72, 75 carbon atoms joined together in this form. So these kind of fullerenes we have. So this is basically an example of C60 fullerene, where you have 60 carbon atoms which are joined to one another, largely in a hexagonal manner. So now fullerene discovery was also important, not only because we discovered the first nanoparticle, but also for its properties. The kinds of properties that we saw with fullerenes were exceptional. We saw that it had very good strength, it had very good conductivity, it had very good heat resistance, it had very good properties of both metals and non-metals at the same time. So all these properties coming together, they are very good radical scavengers as well, that they can be useful against, we'll discuss that later, what is radical scavenging. So we see that there are all these properties that were seen in fullerenes, and that generated even more interest in nanotechnology. That until now we were just theoretically studying a lot of things and trying to come to conclusions, but now we discover a particle, and this particle shows exceptional properties, and that's something that interested the scientists even more in this particular branch. And we see that now the participation of scientists and so that is basically how the things go further. Then we discovered something else and synthesized something else called as carbon nanotubes. Now in case of carbon nanotubes also, if you notice, you still have a hexagonal arrangement, right? You still have a hexagonal arrangement, but this time in a tube like format or a cylindrical form, and that's why we are calling it as carbon nanotube. So because it has a tube like or a cylindrical structure. Now overall, when we look at carbon nanotubes and when it comes to the properties of carbon nanotubes, it was again exceptional properties and even better properties than what we had seen in the fullerenes that we had discovered earlier. That it has high thermal and electrical conductivity, good optical properties, flexibility, increased stiffness, high tensile strength, 100 times stronger than steel when it comes to the comparison, lightweight, and they have the ability to be manipulated while remaining strong. So they can be used in making composite materials, they can be used in various different ways. And that's why today when we look at the applications of carbon nanotubes, we find that because of all these properties that we have seen with carbon nanotubes, this is where they find their applications. They find their applications in various different aspects. So for example, in electronics, we know that they can be used in creating highly conductive wires, transistors, and other electronic components. And at the same time, because of the conductivity that they have, the high conductivity that they can be also used to create computer chips. Even today you have flexible display phones, so for those flexible display phones also we can actually make the use of carbon nanotubes. So in electronics we see that there is a large way in which carbon nanotubes have shown their importance. Then in energy, now in case of energy, they become important because of two properties. That first, they have high or they have a large surface area, and at the same time, apart from the large surface area, they also have high electrical conductivity. They have high surface area and they have very high electrical conductivity, and that's why they can increase the efficiency of energy storage and conversion devices. That because they can store more energy with very high efficiency, and they have very good conductivity, so that's why they become useful in creating any anything related to energy storage. So that's why you can create lithium ion batteries out of it. You can create other kinds of battery technologies. In solar cells they can be used, in fuel cells that can be used. So that's why they can be used in a lot of different ways because of these properties. So that's why in the sector of energy also we understand that carbon nanotubes play a very, very important role. Then after that, in biomedical applications, now in biomedical applications also they can be used because they have good optical properties, meaning that they can be used in imaging applications. So in imaging applications, in devices that are used for imaging applications, they can be useful. Plus they can be used in biomedical devices as well. Making the biomedical devices also, they can become useful, and they can even be used in drug delivery systems. So as we discussed, that what can be done here is that inside the blood cells, because blood cells are of the order of 10 to the power of minus 6 meters, whereas nanotubes are of the order of 10 to the power of minus 9 meters, meaning that the red blood cells are 1000 times bigger than these nanoparticles. So very easily you can fit the nanoparticles inside the blood cells, and you can make the use of blood cells as the carriers of these nanoparticles. So that's when nanomedicine is where they can be useful. You can create medicines, and these medicines can be delivered well to the targeted locations in the body. Even this application has a Nobel Prize. 2016 Nobel Prize was actually given for making molecular machines. 2016 Nobel Prize in chemistry for making molecular machines. So these molecular machines are nothing but the mechanisms that can help in drug delivery with the help of nanotechnology. So even this one application also already has seen a Nobel Prize. Similarly, in making composite materials. Now, because they are very strong and very lightweight. So that's why they can be used in strengthening a material. They can be used in creating better strength in the material. So that's why they can be added to polymers, metals, and ceramics to create strong, lightweight, and heat resistant materials. So that's why they can be useful in a lot of different ways. So that's why composite materials, because we'll be able to reinforce materials, we can add this to the material and increase their strength, reduce their weight, enhance their properties. All these things can be done with the help of nanotubes. Then they have environmental applications, because we can create water filter membranes from them. And again, this is only because of the fact that they have high surface area. So because of that surface area, they can very easily be used as filtration membranes. So they can be used in water filtration also. And at the same time, also to detect environmental pollution, they can be used to create sensors. We can create nanotube sensors to analyze pollutants in any given scenario. Then in aerospace applications also, we see that they can be used in aircrafts and in spacecrafts. They can be useful because they are very strong and very lightweight. So that's why they can help you reduce the total weight of the spacecraft or the aircraft, and that's why they will become more useful uh, in aerospace applications and even in space technology also. So that's why when you look at the list of things that can be done with the help of nanotubes, these were only few things to list, and we see that there is an immense potential that carbon nanotubes have. And that's why in case of the future that we see that there are a lot of ways in which carbon nanotubes will be able to help us even in the future. And whatever we are doing right now, maybe it's just the beginning. As I just told you, that whatever we have achieved in nanotechnology is nothing with regards to what we can achieve with nanotechnology. Now this question was asked in your 2020 prelims examination. Please read the question. Right? I'm not launching the poll for you in this question because there is something that I want to discuss. But can you tell me in comments what do you think the answer should be? Hmm. So lot of C's and lot of D's and some A's and B's. Okay. So let's discuss this. With reference to carbon nanotubes, consider the following statements. First statement says they can be used as carriers of drugs and antigens in the human body. We just discussed this, that yes, we can use them as carriers of drugs and antigens in the human body. So that's why this is fine. Second is they can be made into artificial blood capillaries for an injured part of the human body. Now, have we done this until now? The answer is no. We haven't done anything until now where we have been able to create an artificial blood capillary for injured part of human body. But there is something called as tissue engineering. We discussed this in biotechnology chapter also. Now, in tissue engineering, nanotechnology plays a big role where we are trying to create artificial tissues and organs. So it is an active field of research where we have been trying to see whether, let's say, accident related or any disease related, if there is something that needs to be done. In that case, tissue engineering is an active field of where a lot of research is happening, and nanotechnology plays a big role there. Now, the question or the statement says they can be made into. Now, theoretically, it is possible. Theoretically, it is not impossible. Have we done it until now? The answer is no. But is it possible to do? It should be possible to do because we understand how nanotechnology works and how nanomaterials work, or rather, nanotubes work. So, that seems to be a possibility in the future. And the statement says they can be made, because if you mark this as wrong, that simply means that they cannot be made. So that they cannot be made should not be correct, because uh, you have to leave a scope for the future, that what if in the future we are able to do it. Let's say if it was, the statement says they have been made into, the answer was completely wrong. The statement was completely wrong, that we have not done it yet. So that's why we'll hold on to this. Third is they can be used in biochemical sensors. Now this is already done. This actually has been done. They have been used in making biochemical sensors. So from that perspective, we can say that okay, fine, they can be used. So three is correct. Fourth statement says carbon nanotubes are biodegradable. Now this is where again problem lies. Actively, we have been trying a lot of different methods to try and make carbon nanotubes biodegradable. For example, there are various different types of microorganisms which are being created, genetically modified microorganisms that should have the capacity to eat upon carbon nanotubes. But otherwise, carbon nanotubes are very strong structures, very stable structures, and they are not easy to break down. For destroying carbon nanotubes, we have to heat them to a temperature of more than 800 degrees Celsius, a process which is also called as incineration. If you want to uh, break them, you have to use the process of incineration to break down carbon nanotubes uh, into this. Now, when we say biodegradable largely is associated with the fact that wherever we are using some natural agent for degradation, so then we say that something is biodegradable. Let's say in case of plastic or the polythene bags. In case of polythene bags, we have been saying that polythene bags are not biodegradable, and now we have certain types of plastics which we have started making as biodegradable. But overall, we know that they haven't been biodegradable, and there is only a class that has been made specially to be biodegradable. So so that's why when we say that something is biodegradable, we need to understand the meaning of biodegradable, that what do we mean by biodegradable. Something that will be degraded naturally, something that can be degraded naturally, can be called as biodegradation. Now, in case of carbon nanotubes, natural degradation is not that easy. It's not going to happen in nature. So again, I have a problem with this statement, that I ideally I wouldn't say that they are biodegradable. If again here, if they had said carbon nanotubes can be biodegradable, I could still imagine that, okay, fine, let's mark it, because there are microorganisms that we are making, there are certain enzymes that we are making, so there are ways in which we can actually make them degradable. But saying that they are biodegradable naturally wouldn't be correct. Now, so that's why if somebody asked me what the correct answer should be, I would say the correct answer should be 1, 2, and 3. Unfortunately, there is no 1, 2, and 3. In the absence of 1, 2, and 3, the second best option perhaps could have been 1, 2, 3, and 4. But the answer, the official answer is even not even 1, 2, 3, and 4. The official answer is 1, 3, and 4. Official answer given by the UPSC is 1, 3, and 4. I don't agree with this answer. I would say the correct answer should have been 1, 2, and 3. First and foremost, it's a very vague question to ask. I am not sure why would you ask a question like this, because not even it's not even that you've asked current affairs. There's no current affairs in this. They've just picked up some random data from various papers that are being published here and there. How do you even imagine that a student and an aspirant should know what the papers are publishing? Scientific papers cannot be read by even scientists. It's very difficult to read any scientific paper, and they have just picked up information from scientific papers in this particular question, which is wrong. I mean, I don't even understand why would you give a question like this, but they have given it, and that too with very vague statements that whether they can be made into artificial blood capillaries or not. How do you imagine that a person, uh, forget about non-science background students, even science background students do not know this. You do not know whether they can be made or not unless you read a scientific paper on it, a journal on it. It's impossible for you to know. But they gave this as a statement. They should not have. Same goes for statement number 4, that there are so many researches going on. You just go and look at the number of papers that have been published on whether these are biodegradable or not. I just scanned through, I would have scanned through at least 5 6 papers to try and come to a conclusion whether I can say they are biodegradable or not. It's inconclusive, because we know that until now we have to just heat them, we have to heat them to break them down, and there isn't any natural process that helps in breakdown of carbon nanotubes. But they've asked this question. So I would want to meet these people who even think that they should have asked this question. It looks like that somebody who was not even from, let's say, this background, Googled something on nanotechnology, and few results that they got on Google became their statements in this question. It's very unfortunate that they would ask a question like this. Not only because of the fact that they have given a wrong answer, I would say that they have given a wrong answer, but it's not even the fact. Forget about giving a wrong answer. At least give a legit question. Give a question that people can at least think and answer. But this is not such a question. Anyway, let's not continue with this rant. But this is what it is. This is, I would say, this is my answer, 1, 2, and 3. And this is the official answer, 1, 3, and 4. Next up, we have nanowires. In case of nanowires, we are talking about wires which are having very small diameter. Right? They have very small diameter, sometimes as small as just 1 nanometer in size. Now, this is where they can be useful in electronic devices, but also they can be useful in storage, in computer storage, in making, let's say, computer storage of whatever hard disk etc you have. So making that computer storage shrink, you can make the use of nanowires. They have very good magnetic properties, they have very good optical properties, and because of these properties they will be very useful in storage devices, in making storage devices. So that's why they have remarkable optical, electronic, and magnetic properties, and that's why it is hoped that they will be useful in storing computer data. So overall, right now we are using them to build computer chips or transistors in different electronic devices, but we see that there is a future use where we'll be able to also use it in uh, storage of data, and that's why storage spaces might shrink because of the use of nanowires. So that's why we see that nanowires can be useful here. Then apart from this, another very very important material has been graphene. Now, in case of graphene, again something that has a Nobel Prize already. 2010 Nobel Prize was given to the discovery of graphene. Now, graphene again, if you look at it, you'll see that still you have the same hexagonal structure, right? The same hexagonal structure, but this time arranged like a sheet, arranged like a two-dimensional sheet. Instead of a sphere or a tube, this time you have a two-dimensional sheet. Now, graphene, naturally one of the um, substances with the highest conductivity, one of the strongest substances that we have ever discovered. All these properties at the same time: very good conductor of heat and electricity, very strong in its strength, and at the same time just very very thin. Just imagine that they are just one atom thin, correct? Because it's a two-dimensional sheet of carbon atoms arranged in a sheet like format. And the same graphene, when stacked one over the other, it forms graphite. So graphite basically is made of graphene, where you have graphene sheets which are stacked one over the other. And that's why graphite breaks easily, because these sheets, they can slip over one another very easily. And that is what graphene is. Now, when it comes to the discovery of graphene, the discovery of graphene happened in 2004. And this discovery also was a very interesting event, because the scientists who discovered graphene, they were the ones who actually were just playing around with graphene. They were just playing around with graphene, uh, and I mean, not with graphene, with graphite. So they were using sticky tapes, the cello tapes, as you might know. So they were just peeling off the sticky tapes from graphite, and then they were able to isolate graphene out of it. So that's why they were just playing around with tape, and they won Nobel Prize for it. So maybe any one of us could have won that Nobel Prize, but unfortunately, we were not the ones there. They were the ones playing around with graphite, and they got the Nobel Prize. So 2004, they did this discovery, so-called discovery, and then 2010, they got the Nobel Prize. So, so in case of graphene, and it's very interesting that they got Nobel Prize for it, and the scientists actually who got the Nobel Prize, uh, they became very famous for it, and graphene altogether became very famous, also known as miracle material, also called as miracle material, or also called as wonder material. All these names were given, all these names were given to graphene. Now, uh, in case of graphene, why these names were given, or why these phrases are used? Because one of the best conductors of electricity, one of the best conductors of heat at room temperature, strongest natural material known so far, and thinnest compound known with just one atom thickness. So all these properties in graphene at the same time, and that's why we understood that graphene could be the next big thing, because this is going to change the world. Look at the properties that graphene has. Obviously it is going to change the world. So that is where uh, we saw that it became very apparent that graphene is going to play a very big role in the coming days, in the coming years, and a lot of things can be done with graphene. But today what we see is that, uh, as much as we thought graphene would be useful, we didn't really see graphene to take up the center stage as much as we thought it would. Now, overall, when it comes to uh, graphene, what we see is that today, when we look at the use of graphene, they are being used in a lot of different ways, but not taking the center stage yet. That, for example, let's say, to enhance the strength of other material, to make LED lighting more efficient, in using in electronic devices, used in batteries and supercapacitors. They can be used in as a corrosion barrier between oxygen and water. They can be used in water purification. But these are not the things that you would want a graphene to use as, because you are saying that it is the best conductor of electricity, one of the best conductors of electricity. Can we not use it in electrical wires? We can, right? Theoretically we can. So there are a lot of things that could have been done, but why are we not doing it? The reason for that, is that graphene, when it comes to graphene, synthesizing graphene is very costly. Getting graphene is very costly, and that's why it became. We see that this is where we saw that graphene, because one, synthesizing is very costly, and getting all that or making it work in all these scenarios is going to be a very costly affair, very expensive affair, all these scenarios. So that is where we see that this is where uh, they we have not been able to use graphene as much as we wanted. Although in electronics we are using it, right? In electronics we are using it, in making electronic devices, making foldable displays, we are using it. If you've heard of OLED, there is an OLED television, right? So in OLED televisions we are using it. So that's why we see that overall they actually are being used in all these things, and wherever they are being used they are costly. Foldable display phones are costly, OLED televisions are costly. So wherever graphene is being used, graphene is costly. So that's why this is something which is this is something where we see that there are issues that we have, and we need to find ways in which we can uh, solve those problems. Now, overall, with regards to graphene in India, there is now this Graphene Aurora program that also has been launched by the government of India. And this is where they are saying that this is basically to fill the gap between research and development and commercialization of uh, in graphene, so that we can help in also a lot of startups and development of commercialization of graphene technology in India and graphene adoption in India. So for all that, there is a not for profit section 8 company that is being set up, which is India Graphene Engineering and Innovation Center. So that's why we see that this is where uh, we are now starting to take some effect with graphene, because in India we have seen that graphene research has been lacking. We haven't done a lot with graphene. A lot could have been done, but we haven't been uh, doing a lot of we haven't been getting a lot of headway with graphene. But now we see that these kind of things might change the scenario with graphene. So overall, that's why graphene becomes very important from all that perspective. That the discovery of graphene happening itself was a big event, and from there on, it all started taking a shape. There's something also very interesting. I Let me just divert a bit from the topic. Do you know about another Nobel Prize which is called as IG Nobel Prize? Have you heard of this? Andre GM was somebody who actually won the Nobel Prize, and IG here stands for Ignorable Nobel Prize. Every year, apart from the serious Nobel Prize that is given for physics, chemistry, biology, then in literature, economics, etc., apart from the actual Nobel Prize, there is something called as Ignorable Nobel Prize, IG Nobel Prize, where some scientists have done something that doesn't make sense, but they did a lot of research on that, and that's why they are given Ignorable Nobel Prize. Now, Andre GM actually is somebody who has an Ignorable Nobel Prize and also an actual Nobel Prize. So that's very unique about the scientist who discovered graphene. In 1999, he got Ignorable Nobel Prize for levitating a frog, for showing how to levitate a frog, and he got Nobel Prize for that. And then later for uh, discovery of graphene, he again got the Nobel Prize. So, for example, every year these Nobel Prizes, these ignorable Nobel Prizes are given. Do you know what the ignorable Nobel Prize 2023 for education has been given for? Any idea about that? the ignorable or IG Nobel Prize for Education in 2023 has been given to a bunch of scientists who did a research on why students and the teacher feel bored during a classroom. Why students and teachers feel bored during a classroom? They did a research on this. uh, they published a paper on this, and they got an ignorable Nobel Prize in education for that. Similarly, ignorable Nobel Prize for literature in 2023 has been shown has been given to show what happens, how the brain perceives something when you use the same words many times. So these kind of things are also done. So that's why these are ignorable Nobel Prizes. So this is only one scientist. Why Andre GM is important? He was one of the scientists who won Nobel Prize. So he is somebody. Sorry, GEIM is the spelling. So he is somebody who has won both the Nobel Prizes. So UPSC will never ask you this, but you should know. Read this question. This was asked in 2011 because 2010 the Nobel Prize was given. In 2011 this question was asked. Launching the poll. Please mark your answers. Wonderful. 67% of you have given the correct answer. Let me just discuss this with you once. First statement says it is a two-dimensional material and has good electrical conductivity. Good is an understatement. It is one of the best conductors of electricity. So first is correct. One of the thinnest but strongest material tested so far. This is also correct. Discuss this. It is entirely made of silicon and has high optical transparency. This is wrong. Made of carbon. So this is wrong. It can be used as conducting electrodes required for touchscreen LCDs and organic LEDs. Correct. We discussed that in OLEDs we make the use of graphene. So 4 is also correct. So correct answer will be 1, 2, and 4. Then coming to the last topic in this segment, quantum dots. Nobel Prize in Chemistry 2023 has been given to quantum dots. Now, what are quantum dots? Understand what quantum dots mean. So, two terms: quantum dots. Quantum because of quantum mechanics or quantum properties. What are quantum properties? Let's hold on for now. This is also something that we are going to discuss later in another chapter. So, because they have quantum properties basically are the properties that govern subatomic particles. The physics of subatomic particles is nothing but quantum physics. So, these are also very small particles. These are also nanoparticles, hence the name quantum, and the term dot because they are spherical in shape but very small. So from where we are looking at, it's just like a dot for us, correct? So that's why the term quantum dot has been given to these particles. Now the properties of these particles become very crucial because when these quantum dots are emitted with the help of an ultraviolet light, when ultraviolet light is passed through the quantum dots, they emit various colors. And the color is dependent on the size of the particle. Now, what we know is, and something that we are again going to discuss later in IT and communication chapter, that when you look at the visible light, you might know that visible light has seven component colors, right? Vibgyor, as we call them: violet, indigo, blue, green, yellow, orange, red. So we have these seven component colors in white light. Red in this has the longest wavelength, and violet has the smallest wavelength. So that's why the bigger sized quantum dots, they show red color, and the smaller sized quantum dots, they would show blue or violet color. So color is dependent on the size. So bigger size, red, and smaller size, blue. So the size, the color is dependent on size. So because of this, what happens is that they have uh, good optical properties, and we see that they are also useful. So now what, when we look at their applications, they will be useful in photovoltaic devices. They will be useful in making QLEDs. Now you might have heard of this, also is advertised a lot, that there are QLED TVs in the market, especially largely marketed by Samsung, and then there are some other companies also. So in QLED, if you look at the advertisements, the, let's say, Samsung advertisement, so you will see that uh, Samsung advertises that these QLED TVs will give you better color contrasts. They will give you better color reproduction compared to any other normal LED. Why? Because they are using quantum dots. So with the help of quantum dots, they are able to produce better colors, and hence they say that they give you better colors, better pictures, better contrasts as compared to normal LEDs. I'm not advertising Samsung. I'm just saying that this is what they say in their advertisements. So quantum dots become useful there. Then in medical field they can be used, and they are also useful as photocatalyst, where they can become useful in hydrogen production. In hydrogen production also they become useful, and hence we see that they can actually be used here. In medical field they can be used in cancer care. In detection of cancers also, quantum dots are useful. So that's why we see that there are medical applications, applications in electronics, applications in energy, applications in photovoltaic devices, in solar cells. Everywhere we'll see that quantum dots become useful, and that's why the Nobel Prize has been given to quantum dots, because we have seen that there is an enormous potential, and overall quantum dots have been gaining their share in the world. So hence quantum dots, and that's why the 2023 Nobel Prize has been given to quantum dots. So let's end our session here, because the other topic starting now will take a lot of time. Any questions if you have with regards to whatever we have discussed until now? Any material exhibit their properties based on their electrons arrangement in that atom. If we can change the number of electrons in an atom, then that material will that material exhibit different properties? Yes, yes, it would. For example, carbon has 6 electrons. Yeah, that is possible, and this is, I mean, not doable yet, but it is one of the things where we look at the nano research, where if we can change the configuration, if we can change the structure, then we can change the properties. For example, let's say if you take melting point, suppose you have these two materials, let's say there are these two surfaces, and if you are analyzing the melting point of these two materials, melting point of this material will be higher, and melting point of this material will be lower, because all the heat will be concentrated here in this, so it will melt faster. In case of this surface, first the heat will get distributed all across the surface, and then it will start melting. So now let's say if nanotechnology gives us a way in which we can flatten out this surface into this, so then you will be able to increase the melting point. So in that way we can change the properties, and if, let's say, practically if it was possible to change electronic configuration, then what you are saying is possible. I also want Nobel Prize for experimenting with my own life and still living it. You'll have to publish a paper first, and then you get the Nobel Prize. If quantum dots got the Nobel Prize this year, how is Samsung already using it? Nobel Prize is not given for discovery happening this year. Discovery of graphene happened in 2004. Nobel Prize was given in 2010. Last year's Nobel Prize was given for research that happened 20 years back. So it's not that you do a new research and you instantly get that Nobel Prize. No. If that science or if that discovery or invention has seen certain changes in the world, or they have enormous impact on the scientific world, then it will be considered for Nobel Prize. The uh, let's say the research on gravitational waves has been happening for 40, 50 years, and then after that the Nobel Prize was given to it. So it's not that you get Nobel Prize the next day or the same year. Leaderboard, okay, let me show the leaderboard. So this is the leaderboard for today. Congratulations to everyone with correct answers, and there are so many of you. Wonderful. And others need not worry. If you have understood what went wrong for you, you are good. Can you please repeat Terminator genes? Terminator genes basically are genes that which are artificially inserted in a seed so that the seeds germinate only once. They cannot be used multiple times, or the seeds which you get out of the fruits of that plant cannot be used in the next season. Nanoviruses, optical fire. No, 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 no. Completely different. Completely different. How quantum dots work? Basically, there are various mechanisms that we use depending on what kind of application we have. So there are a lot of ways in which it can work. About the next class, about the remainder of the classes, I'll just check with the team when it can be done. We'll announce it in advance accordingly so that I can conduct the classes whenever possible. Is there any new notification for prelims? The notification has not come out yet. We are still waiting. See, graphene was discovered in 2004. I'm not saying that graphite was discovered in 2004. We have known about graphite. We just did not know that graphite is made of graphene. That discovery happened in 2004. What is A, B, and Z DNA? So basically, in terms of DNA, it depends on sometimes the thickness of the DNA. That is something that varies. Base pairs, sometimes the way the combination of base pairs happens, that varies. So that brings in variations, and that's where there is A, B, Z, AB, AZ, all these kinds of DNA. But that's not required for your examination. That's way beyond the syllabus requirements. Do we have to make figures? No. I mean, if these are small line diagrams, you can, but the complex figures you don't need to draw. On Monday, I don't think I have my class because I think there is some other class which is starting for you. But we'll see. Whenever it is possible for us, we'll notify you in advance and we'll conduct those remainder of the classes. Tomorrow and day after, I am having the weekend class, and weekend class almost is running parallel to the weekday class also. So only, I mean, because I miss certain things, so maybe the second half of Sunday will be some extra topics compared to what we have completed in today's class. So if you want to come there, you are most welcome, you can come there. When one person resembles other, what aspects the resemblance for this? Aspects of various traits and various characters that one has, the genes that people have. So that is what becomes the base of all the resemblance. Right then. So let's end the class here, and let's please go back, revise everything. Any question if you have, you can always bring it back. You can write to me, you can send it to me. I i once again apologize for those of you I haven't been able to respond to, but I'll definitely do that. rashesh.kumar@byju's.com. All right, so this is the end of the discussion today. Thank you.